What a spooky, scary season, everyone. And how better to start off such a very scary October than with a funeral? My dearly beloved, gentle and modern apes, today we say goodbye to my patience. Unfortunately, patience, as it turns out, is very fragile. As fragile as, well, the light of a candle under the breath of a stranger. You know, these things were actually supposed to be blood candles. I got them for like $2 at Walmart, and instead of being bloody candles, they're like pink. You know, it's like a pink color. I'm not very happy. I would leave a bad Amazon review if I had procured them on Amazon, or if I, felt like reviewing something on Walmart. But we will use these candles because they are indeed very spooky and reminiscent of the season, and they are a nice tribute to pay to my dearly departed patients. So let's talk about that. So as anyone who's been following this channel since its inception will know, I kind of make it my business to engage with any and everyone who has any questions whatsoever about evolution, specifically human evolution, the age of the Earth, the whole big shebang. And that's partially because I find the study of all of those subjects to be humbling, grand, and fascinating. It's why I buy t-shirts with like the stuff on them. I genuinely love it. I love having the conversations. And I also enjoy it because once upon a time I was a young Earth creationist myself. So being a good faith actor is changing your mind when you're proven wrong, or at least changing aspects of your line of thinking when shown that the data says otherwise. This doesn't mean you have to abandon the whole shebang, right? Like in the case of creationism. You don't have to just throw it all out, although you probably should. You just need to tinker with your model a little bit when the data shows its ugly face, right? But God, I've met so many people who just double down on the same exact points when shown that the points are faulty or flawed or false. And that just does not feel good faith to me anymore. It really doesn't. If you hold to a position, despite the fact that there exists very solid evidence to the contrary, and you don't even change the opinion a little bit, well, you're rigid and you're kind of foolhardy, and in my opinion, you've kind of become unreachable. And thus you cease to be worth the time of both myself and basically my entire audience, and anyone who really kind of, gives a shit about empiricism and, truth and understanding the world around us. You're a bad faith actor at that point, and you're not really a lot better than the flat earthers. So if you can't tell, a lot of this isn't really scripted. A lot of this beginning part isn't scripted, and that's because I'm trying to keep the parts that aren't pure data more personal, because this is kind of a personal issue to me, and that's because I'm taking some kind of intense steps in this video, because we're going to be analyzing why, for the time being, in my opinion, Standing for Truth's channel, and thus Standing for Truth himself, is a bad faith actor. And unbeknownst to me, up until this point, probably always was. This is a long video. It took a lot of research, and it took a whole lot of time, and I've got a couple of guests too, and I recognize this isn't the type of video that is going to be everyone's cup of tea, and you may want to just zone in on specific topics that I'm covering. And so, with so many concepts, I decided to include a table of contents, so to speak, because there's not going to be an abridged version this time. So, see the table of contents in the description, and then right now. Okay, so, problems. I like standing for truth, and I think that standing for truth is a bad faith actor. What the hell do we do with that? Well, what we're going to do is we're going to give him the tools to show that he's a good faith actor. That's what this video is going to be for. We are going to address the problems, the reasons why we, and by we I mean I, but you're my audience, so you have to think what I think that he's acting in bad faith, and then we're going to present the solution to the problem. And spoiler alert, the solution is just answering the dang questions that I give you. You know, that way we've got a two-way street. But first, we have to talk a little bit about the nature of the problem, the pathology of the situation. We must diagnose it like powerful doctor scientists. It's so silly. So what's worse than a bad faith actor? A bad faith actor pretending to be a good faith actor. And the latter are pictured here.
I'm a stupid moron with an ugly face and a big butt, and my butt smells, and I like to kiss my own butt.
The latter are very, very difficult to diagnose, and it's dangerous to do so because you really can never truly know someone's intentions, which is why you shouldn't say someone is a bad faith actor. You should really say that you think that they're a bad faith actor. It is in your expert opinion that they are indeed acting in bad faith. So if you're here, and I assume that if you are, you know me pretty well by now, you should know that it's kind of my gig to try and give people the benefit of the doubt and assume good faith unless there's some damning evidence to the contrary. For example, Nathaniel Jeanson, creationist author of Replacing Darwin, is, in my opinion, the quintessential bad faith actor pretending to be a good faith actor. He has a PhD from Harvard in a STEM field, so we know he's taken a physics course or two, perhaps in undergrad. However, in his book, he justifies using a constant mitochondrial mutation rate in order to get his 6,000-year date origin for humanity by pointing to the fact that since physicists use a constant rate for the speed of light and nuclear decay, why can't he? Jeanson is presumably not a brain-dead moron. He knows the difference between the constant rate in physics, dictated by the Stoic laws in math, and rates in biology, which are subject entirely to their environment. That's covered in my Physics 1 textbook. Now, in my brain, it's always good faith hours. So maybe Jeanson is just an idiot in every field besides his own. But then I saw that Jeanson has also admitted he only wants to use his doctorate for furthering his religion. Bad faith. That is why, for the duration of this video, I will be referring to the act of bad faith acting as Jeansoning. You see, I am concerned that the frontman of the SFT YouTube channel, Standing, is Jeansoning. He is asserting the same points despite evidence to the contrary, and he's doing so without actually presenting reasoning to why this evidence should be invalid, disregarded, if you will. Allow me to introduce the crime.
What are you gonna do to us?
Okay, follow me. If you can't do the time, don't do the crime. Okay, time's up. Now get out.
But we...
Misrepresented science.
Yeah, on
The internet.
That being Standing's most recent YouTube video to me, yours truly. I'm ever so pleased with the copied clickbaity title, but flattery will get you nowhere, Mr. For Truth. You've been charged with the crime of Jeansoning, the punishment for which is no further engagement, banishment, if you will. That means no more conversations or debates or direct responses to any videos you might throw my way. Now, you might be asking, Erica, my favorite local Gutsick Gibbon, why are you making this move now? What is suddenly so pressing that you have to cut them off sort of at the source? Don't you like busting creationists? And the answer, dear viewer, is yes. I love busting creationists. I get immense joy out of it. I find it to be an educational experience for both myself and my viewers, as I do the necessary research to pick apart each individual facet and sinew of their argument. But this channel is run by one person, me. I do all the writing, I do all the research, I do all the editing, and I'm also trying to slog through my thesis at the exact same time, which means I don't have time on my own to dig through every 2 to 6 hour video response to me that actually says nothing of merit throughout its entirety, or at least very little compared to the challenge that was posed to them. Part 2, the diagnostic characteristics of an SFT video. Now we're going to go through some of the typical tropes that we tend to see in videos put out by the channel. The first is repeating arguments they have been corrected on, while providing no reason as to why they are indeed correct, and the criticism is wrong, actually. Human chromosome 2 is an excellent example of this, as SFT has yet to respond to my breakdown of alphoid repeats, instead repeating Tompkins tropes. Number 2, using words like refuted, debunked, overturned, discredited, and exposed when referencing scientifically robust concepts that are currently accepted in the field. This is usually combined with confidence adjectives like definitely, completely, absolutely, or entirely. Number 3, selective trust in experts, depending on whether or not they can use a work of said expert to support an opinion they hold. This would be like the recent appeal to the work of Ella Bin to justify the presence of 2 hominins at the Malapa site in order to hand-wave away Australopithecus sediba, but then seemingly ignoring the work of all other paleoanthropologists on the subject. Number 4, ad hominems, depending on whether or not they like the person that they're trying to review. Number 5, frequent misunderstandings or misrepresentations of concepts such as fitness or selection. And when the understanding is present, they literally try to redefine them. See Stanning's video that attempts to redefine fitness in order to grapple with it. Next up is an utterly incomprehensible understanding of how literature works, such as why consulting up-to-date literature is so important, and when using older papers can be acceptable. For example, you can look at almost every video. They are chock full of ancient quote mines applied to constantly changing fields like genetics, papers on subjects with more up-to-date material, and appeals to pop sci articles and non-published works. Much of the above can be chalked up to the fact that none of the members of the Standing for Truth Brain Trust have actually had formal education in the subjects that they so frequently discuss and assert with authority. This would be biology, evolutionary biology, genetics, paleontology, paleoanthropology, geology, physics, chemistry, and the like. This lack of education is especially stark in the videos where the Brain Trust attempts to shame people, i.e. me, for calling in experts in the field that I am not formally educated in. That's how this works. I'm not going to speak on genetics as if I'm an expert if I don't have time to do all of the research myself, or perhaps if I simply don't understand the concept to the degree that may be required to, again, tease apart the minutiae of these arguments that they and Nathaniel Jeanson put forward. My interactions with the Brain Trust via video tend to go a little something like this. Wrong footage. They tend to make a claim in a video or debate that is absolutely asinine, such as hydroplate is a viable flood model. I respond in a heavily edited and sourced video that systematically blows that hypothesis the fuck out and ends with an organized and straightforward direct series of questions or challenges that must be addressed. Then, the trust will either shut up about it or respond once more with a video that either handwaves or ignores entire points of the response that I made, but the video will be titled as if it is something all-encompassing. Let's look at 2 examples. In the case of the heat problem, I didn't hear from Standing in a direct video response. Originally, this was in the shut up about it category. But then, in a more recent video, Standing offhandedly mentions that there is no heat problem and suggests his audience look into videos of his that he has linked in the description that apparently address it. videos that came out before my heat problem video, thus could not have and indeed did not actually address the problems. In the same breath that Standing is encouraging folks to check out his videos, he confidently asserts that even if there was a heat problem, it just doesn't matter because rocks show accelerated decay anyways, and that proves a young Earth. Now again, the big creationist Ultra Chad Alphas, Answers in Genesis, Institute for Creation Research, and Creation.com all openly admit that there is in fact a heat problem, and it's currently being looked into because, short version, when elements decay, they release heat. And since rocks are made up of elements whose decay rate suggests a 4.5 billion year old Earth, cramming 4.5 billion years of heat into a 6,000 year time period kills everything and boils off the oceans a few times. So, they're looking into it. The only person not doing that is Walt Brown, who is coincidentally the hydroplate guy. Whose model would vaporize the granitic crust of the Earth a few dozen times. I imagine this probably gets pretty awkward at the creation conferences. I mean, if Walt is even invited to those. And now the Creation Network presents Afternoon Yek. Hydroplate.
Cancelled.
Standing won't outright say who he supports, but he basically has two options: support the big guys and admit that there is a problem, or support Walt and answer my posed questions. He has done neither. Instead, he has just said that there isn't a heat problem and moves on to cite a bunch of wildly outdated reasons as to why some creationists from the 80s and sometimes in the early 2000s, suggest that the rocks indicate accelerated nuclear decay. This would be an example of Jeansoning. Now, prepare yourselves for the response on this one. I suspect that the brain trust will essentially hear what I have to say about this insurmountable problem to their model, if you will, and then they will gesture angrily and wildly to abiogenesis, which would be perceived as an equal problem for evolutionary theory, because of the nature of it being a question that isn't fully answered by secular science. But there is an important distinction between the two. In the case of abiogenesis, there are aspects of the process that have not yet been answered, a lack of current evidence. In the case of the heat problem for a 6,000-year-old Earth, there is active evidence to the contrary. To compare the two would be false equivocation. It should come as absolutely no surprise to anyone keeping up to date on this particular issue that these objections to the ancient nature of the Earth were blown the fuck out 15 years ago. At the latest. Standing kind of seems to get his feelings hurt when I get a little personal on this kind of thing, when I really roast these guys, not Standing, but his sources, for bad methodology, weird conclusions that they reach that involve a ton of presupposition and weird ad hoc reasoning. And I'm sorry that kind of hurts his feelings, or at least seems to hurt his feelings. But the nature of this is that it's pretty cut and dry, and I don't know what kind of support Standing is propping up for this accelerated nuclear decay and for a solved heat problem. That being said, we're going to zip through some of the objections to radiometric decay now. We'll likely go through them more in depth at a later juncture, but, I like to be thorough. But before we begin, we should probably cover what actually makes a good natural clock. The first of the criteria for a good natural clock is that it has to have a known initial condition. A natural clock must also have a process with a constant rate with which measurements of age can be made. The other two requirements for a natural clock are that the process of the clock must be irreversible and it must have a known final condition. So what is radiometric dating? How does it work? Does it fit the bill? Radiometric dating works like this. It is based on the law of radioactive decay, which recognizes that some atoms of a particular chemical element spontaneously change into atoms of a different particular chemical element. The parent element is reduced to a daughter element and an alpha particle. Only igneous rocks can be used as reliable natural clocks as they have a known initial condition, all parent and no daughter. This is simply a fact of geology. When igneous rocks form, they are indeed all parent material. We also know that the rate of decay is constant, meeting the second criteria for a natural clock. Well, how do we know that it's constant? Because we've literally thrown everything and the kitchen sink at the process to try and speed it up. We've tried high and low temperatures and pressures, irradiation with X-rays and gamma rays, bombardment with high-energy particles, various levels of magnetic and electric fields, and incredibly high accelerations of gravity. Conditions which would never be seen in nature on our planet. And the best we could do was acceleration by 1.5%, and even that was on a geologically irrelevant element. So the rate is, to our knowledge, constant. This is something that was admitted by the very creationists who were trying to prove that it wasn't. Decay also has a known final condition, that being all daughter material, and the process is indeed irreversible. Radiometric dating is also corroborated by a dozen or so independent methods as well, and the fossil fuel industry relies on its accuracy for finding said fossil fuels to the tune of around $250 billion annually. So this seems pretty solid. What are the objections? First up, fission tracks. Fission tracks as physical structures are simply linear tracks in rock crystals usually around 6 to 10 meters long. Fission tracks are most often caused by the spontaneous fission of the parent uranium-238 atom into two daughter atoms of polonium-119. 238 uranium is well documented in radiometric dating records with its decay into 206 lead with a half-life of about 4.5 billion years. Fission track dating has a very good initial condition, being there are no fission tracks evident in newly formed rock. This is, of course, observed and confirmed today. Fission track dating also confirms the constancy requirement, as the spontaneous fission of 238 uranium atoms occurs at a constant rate under natural conditions. The spontaneous fission of 238 uranium is irreversible, as there is no known process in the universe that can fuse two polonium atoms together. The evidence of fission tracks, however, can be considered somewhat reversible, as they easily disappear with heating. This is why fission tracks can only measure the last cooling of the rock, not its age of formation. Fission track dating has a known final condition. When all of the 238 uranium atoms undergo fission, the constant rate of the clock will cease and no new tracks will appear. The RATE team, a crack team of creationist geologists attempting to disprove radiometric dating, decided to take a crack at it. So the RATE team decided to take a bunch of zircons and other minerals from all across the western United States in order to test whether fission track dating fell in line with the rest of the radiometric dating methods. They basically concluded, in a paper authored by Andrew Snelling, that some of the results for the Middle Cambrian samples, quote, failed to agree with previously published results, showing major disagreement or discordance, De Young 2005. The research team then explained that the discordance was probably related to the thermal histories of the rock and differences in measurement technique described by Snelling in 2005. Furthermore, De Young discussed the regional history from the area where the samples were collected and asserted that some of the discordance could be attributed to the fact that tectonic movement in the Colorado Plateau long after the Middle Cambrian caused the rocks to be heated above their annealing temperature, and the clocks were thus reset. The ICR team went on to state that almost all other results are concurrent with dates previously obtained for the rocks using other dating techniques. From this statement, ICR admits to believing that millions of years of decay seems to have occurred in these rocks assuming a constant rate of fission. ICR appears to have followed standard procedures and obtained typical results from the rock samples they collected, as one cannot find any serious flaw with their methodology or the results which they obtained. And yet, here is ICR in the year 2020 touting exactly the discordant dates and nothing else. ICR is Jeansoning here. Because here's the rub, right? The very authors cited by ICR in this paper propose reasonable natural methods for why they got a handful of discordant dates, and they further admit that the majority of the dates corroborate typical ancient Earth measurements. So what the hell, ICR? Fission track dating is also corroborated by like a dozen other radiometric dating methods. Oopsie. So the consensus on the entire affair of using it as a corroboratory method of dating rocks seems to be, by the admission of ICR's data as well as countless other analyses, that it works just fine. Next up, radiohaloes. Radiohaloes occur in certain types of igneous rocks such as granite that contain minerals like zircon and monazite, which can be inclusions within other minerals such as mica. It is known that the crystal lattice of these minerals commonly contains traces of certain radioactive elements. These radioactive materials can leave radiation damage in the form of discoloration in the surrounding rock. This radiation damage, or halo, appears as a fuzzy, spherical-shaped discoloration in the mineral structure emanating from the location of the radioactive material. Creationists and young Earth proponents use a specific type of pleochroic halo, purported to be caused by the radioactive element polonium, to make the claim that Earth could not be billions of years old, but must be much younger. It is important to note, however, that polonium halos do not give any specific age of the Earth, and young Earth supporters are not attempting to use them as a dating method. Instead, polonium halos are seen as a way to discount scientists' claims that the Earth has a geologic record which took billions of years to form. In short, polonium halos are not natural clocks because they cannot be said to give a specific age, but creationists infer certain things about these halos that they say is evidence of a young Earth. So polonium forms from the alpha decay of radon, which is one of the decay products of uranium. Since radon is a gas, it can migrate through small cracks in the minerals, leading to the polonium halos. The fact that these halos are found only in association with uranium, which is of course the parent mineral for producing radon, supports this conclusion, as does the fact that such halos are commonly found along cracks. When microfractures are not seen, it always seems to be when the crystals were formed in magma, which doesn't tend to crack at all. Additionally, if polonium halos truly had a nearly instantaneous origin, as suggested by Snelling, then even more examples of other polonium halo types would be expected to occur. We should see halos from 215 polonium and 211 polonium, as well as from 216 polonium and 212 polonium, but these are not found. The reason is that the radon gas atoms in these two decay series, which are the precursors for other radioactive polonium isotopes, have half-lives in the seconds, and their daughter polonium isotopes have half-lives in the seconds and microseconds, instead of around 3.5 minutes for 218 polonium and 140 days for 210 polonium in the 238 uranium decay series. However, Gentry only found the one kind of polonium halo sequence among the three possible kinds in biotite and fluorite, of supposed instantaneous origin. So basically they had an opportunity for a prediction here, but the prediction was not met. So it looks like this one is busted as well. Bummer. Now we can talk about helium in the crust. Some creationists claim there is too much helium in Earth's crust for Earth to be any more than 2 million years old. If Earth has existed for billions of years, there should be little helium left in the deeper rocks as a result of radioactive alpha decay. They claim that if God had created the Earth with the initial helium in the atmosphere, the maximum age would be even lower than 2 million years, perhaps even as little as 6,000 years. The RATE project, which was of course co-sponsored by the Institute for Creation Research, the Creation Research Society, and Answers in Genesis, claims that the amount of helium present in minerals at different depths of the Earth's crust is simply too high to support the day age or evolutionist theories of an old Earth. They sent rock samples to a lab for helium diffusion tests, and their results were that the rock samples have too little resistance to the diffusion of helium through the rocks for the age to be greater than at most 2 million years. The conclusion reached by the RATE project as to the reason for the increased helium is that sometime in the past few thousand years there was a period of increased radioactivity. A fundamental problem with this hypothesis, however, is that the amount of energy released during the accelerated decay proposed by the RATE team would potentially be enough to evaporate the oceans and melt the Earth's crust. I wonder if Standing has considered this when proposing this flaw in radioactive decay. Likely not, as we have yet to hear from him about the heat problem. Not only that, but subsurface pressure and temperature conditions affect how quickly the helium diffuses out of zircons. Humphreys et al. Selected a rock core sample from the Fenton Hill site, which Los Alamos National Laboratory evaluated in the 1970s for geothermal energy production. The area is within a few kilometers of the Valles caldera, which has gone through several periods of faulting and volcanism. The rocks of Fenton Hill core have been fractured, brecciated, and intruded by hydrothermal veins. Excess helium is present in the rocks of the Valles caldera. This helium may have contaminated the gneiss that Humphreys et al. Studied. In short, the entire region has a very complex thermal history. Based off of the oil industry experience, it is essentially impossible to make accurate statements about the helium diffusion history of such a system. So why choose volatile rock formations known for giving bad data? This would be like choosing metamorphic rock and using dates from that to attempt to disprove radiometric dating, which requires an igneous rock. So it looks like helium doesn't really fit the bill either. So what about magnetism? This creationist argument is based on the theory that was proposed by Dr. Thomas Barnes in 1971 and then again by Humphreys in 1993. Using data obtained by McDonald and Gunst in 1967, Dr. Barnes asserted that Earth's magnetic field has been decaying in a non-cyclic manner at an exponential rate since the beginning of creation. Barnes used the data of McDonald and Gunst to plot an exponential curve and, by extrapolating the observed data backwards in time using his exponential decay equation, Barnes claimed that the magnetic field was approximately 40% stronger in 1000 AD than it is today. Continuing this extrapolation, Barnes stated that the Earth must not be older than 10,000 years, or else the strength of the magnetic field would have been so large that it would have melted the Earth. Unfortunately for Dr. Barnes, observed scientific evidence has shown that the Earth's magnetic field has not been decaying constantly since the dawn of creation. In fact, the magnetic field has fluctuated and reversed in polarity over time, proven by evidence of periods of increasing and decreasing field energy. Due to this cyclic fluctuation, scientists have argued that the strength of the magnetic field cannot be used to determine the age of the Earth, since it is, in fact, a reversible and cyclic process. However, creationists have attempted to offer a solution to this problem. Dr. Russell Humphreys, a nuclear physicist, looks to the Genesis flood as the cause of the fluctuations in the magnetic field. Utilizing a creationist theory that the Genesis flood was caused by the plunging of tectonic plates towards the Earth's core, Dr. Humphreys claims that the tectonic plates would have caused a sudden cooling in the outer parts of the Earth's core. This sudden cooling, he says, would have caused the convection currents to flow within the core, which would generate numerous reversals of the magnetic field over the course of thousands of years. These field reversals would have happened rapidly at the time of the flood, according to Humphreys, leading to an increase in the intensity, strength, and direction of the magnetic field until it reached a maximum at the time of Jesus Christ. The scientific community has been quick to respond to the age data presented by creationists, and they have provided several counters to the claims made by Barnes. Any flaws found in Barnes' work also discredits the work of Humphreys, since Humphreys' work is based upon that of Barnes. The first made by scientists is that Barnes used an outdated model in his analysis of the Earth's interior. By using an outdated model, any assumptions made by Barnes become instantly invalid. Second, by using McDonald's and Gunst's data, Barnes only analyzed the dipole component of the magnetic field, which is not an accurate measurement of the overall strength of the magnetic field. Third, scientists showed that the data used by Barnes more easily fits a linear curve than an exponential one, and Barnes chose the exponential curve based on incorrect assumptions. The scientific answer for the apparent decay of the magnetic field was given by Dr. Walter Elasser, a physicist at the University of Utah. According to Elasser, Earth's magnetic field is generated by a dynamo within the Earth's core. This dynamo used to be heavily understudied, leading to creationist criticism. But time keeps on slipping, and we find out that the dynamo theory is in fact robust. Magnetism ain't gonna cut it. At long last, we reach cold material, which is Baumgartner's theory, and it still doesn't work without miracles, as Baumgartner himself admits. The thermal diffusivity of the Earth would have to increase 10,000-fold just to get the subduction rates that he proposes, and something would have to cause the advance and retreat of the magma bubble. Miracles would also have been necessary to cool the new ocean floor and raise sedimentary mountains in months rather than the millions of years it would ordinarily take. Baumgartner himself estimated a release of 10 to the 28th joules from the subduction processes alone. This is more than enough to boil off all of the oceans. In addition, Baumgartner postulated that the mantle was much hotter before the flood, and the heat would have to go somewhere. So these objections to radiometric dating are pitiful and outdated, which has me wondering, why is Standing using them? Why can his channel not just admit that they have a problem? In the case of Homo habilis and Australopithecus sediba perfect mosaics, I did get a lovely, long, drawn-out video that was essentially a fetid regurgitation of contested bones arguments. And better yet, when I took the time to really get down and dirty with the literature, that is to say, I walked the Brain Trust and the rest of the audience through half a dozen or so papers looking at the minutia of each and every bone that made up these two hominins, I got this. Then why confidently assert in the first place that these hominins were bogus, especially when neither you nor the two authors of the primary source that you use have any formal education in the field? The weird thing is, I've gotten a couple more videos by him, approximately 6 hours worth of content that I've gone through as well, as you've already seen. At least you should by now. I should have published that prior to this. That's the plan. You know how scheduling goes. And they're still not really saying anything. They're still not answering the questions. There's just a whole lot of, gesturing to these big concepts that I'm not 100% sure Standing understands the full magnitude of. To my knowledge, Standing for Truth doesn't actually have a human evolution textbook, and instead opts for a short handbook written by Bernard Wood and a pop-sci book with some lovely visuals. I have it behind me. It's called Human Evolution, or Evolution the Human Story, I believe. I think you might not be able to see it. I'm not going to pull it out. And both of those are lovely, but they're no substitute for legitimate literature or a textbook on the subject, which is going to give you the most up-to-date information, and it's going to do so in a way that's been peer-reviewed by countless paleoanthropologists. On a more personal level, I have a bit of a beef with Standing for Truth and the rest of his gang, his ilk, if you will, in how they've treated some of my buddies here on YouTube. Those issues would essentially be that Standing is seemingly afraid to or won't talk to or debate Jackson Wheat, who's frequently asked for a conversation from the guy. Standing won't take Dan Stern-Cardinal's, Dr. Dan actually is what he's known by here, his evolution challenge, which is essentially just proving that you understand the basic concepts in evolutionary biology. To my knowledge, Standing agreed to it and then backed off. I don't know why he did that. And lastly, Standing kind of keeps the company of a couple of real rude folks, and I'm not that big on that. But now that we kind of have a good background on the nature of what a Standing for Truth video encompasses, let's get into Part 3, The Last Straw. For me, it's always been easy to deal with Standing for Truth. I've enjoyed all of our interactions in the past, but upon watching some of his more recent videos, something has become abundantly clear, and that's that he doesn't plan on answering any of the questions that I pose his way, or at least he doesn't plan on answering or even alluding to the vast majority of them, and I'm really starting to grow frustrated. I'm going to address some of the main points from the video, one of the videos, this video here, and then, depending on how mad I'm feeling at Standing when this is being recorded, we may or may not go into how he went a little bit apeshit in the comments section.
We're leaving no stone unturned. Matt, good to be here, brother. How you doing tonight?
Doing good, man. Thanks. This will be fun.
This is going to be a lot of fun.
Now this video is very long, so I've taken the liberty of clipping out some of the more important bits. You can find the original in, of course, the description. Now let's begin with listening to what I consider to be Standing for Truth's channel thesis statement.
At the end of the day, and it's funny, Matt, what are your thoughts on this? Genetics is what's most important.
Let's put a pin in that and commit it to our memory, shall we?
Remember early on when they built their phylogenetic trees before any genetic data was in, and then all of a sudden the genetic data came out and the entire tree, they all changed? Remember that? It was like, right. That's because of how far off they were with assumptions.
First and foremost, no. The tree of life did not change very much once genetics was introduced. Some of the terminal branches may have swapped around here and there, but for the most part, it stayed pretty constant. In fact, it was surprising to some how close morphology had gotten people.
We've gone over that, which is that can kind of differentiate between humans and chimps. For example, we discussed the similarities, but the fact that in our genetics, the similarities oftentimes have incredibly different gene expression roles and functions. So we're not gonna, rehash that, but definitely check out that video.
First of all, notice how vague he is. What portions of the genome are expressed differently in humans and chimps specifically? The thing is, there are absolutely differences between the two species, otherwise humans just would be chimps or vice versa. But, and this is going to shock you, if you use Standing's method here of expression differences being what separates humans from other apes, just like every other metric creationists try to use, you end up with an absolutely insane amount of biblical kinds. This is not a method that can be standardized efficiently across the entire animal kingdom, leaving them once again stuck with no way of separation.
So when we actually look at animal kinds, we can see that there's oftentimes a lot of diversity, let's say within, the felids, for example, which is what we would expect. So she tried to debunk that by pointing to specific species that have experienced bottlenecks, whether it's due to inbreeding. There's a number of reasons that can reduce genetic diversity. Look at the cheetahs. The cat family in general has high levels of genetic diversity, because her question to G-Man was, if species within the same family that we say is the same kind, say the cats, the felid, some of them have more DNA differences and DNA diversity separating them, yet we as creationists conclude they're the same kind. Then humans and chimps, she'll say, have less DNA diversity, DNA differences separating them. So why would we conclude two cat species are of the same kind, yet they're separated by more DNA differences, and not conclude that humans and chimps are? Well, like I said, we'd expect the lower levels of genetic diversity in humans, more in the animal kind. So she completely failed right off the bat on one of the 0.1% things that she tried to actually touch on, by pointing to a species, believe it was a gorilla one. I'm going to leave the video in the description. Actually, you can see it in the slide before this.
You've missed the point entirely.
I don't know where standing draws the line within the ape kinds, as some creationists do have each hominoid as its own biblical kind, but even if Standing for Truth tries to use genetic diversity as his great separator of kinds at the family level, he runs into problems, because there are entire families of animal with lower genetic diversity than just humans. So still, no standardization, guys.
What are your thoughts, Matt? And then we can move on, brother.
Sure. I was just thinking about it from a logical perspective. If humans and chimpanzees actually share a lot of these similarities, and there's only a few substitution differences between us, but yet there's more differences between dogs. Well, wait a minute. Shouldn't there be many more differences if we split from these 6.5 million years ago? I mean, if you literally watch a new dog breed come into existence and it has more substitution differences than the nearest breed that it comes from, then obviously time isn't the factor. So I just find it pretty ironic then that they can say, oh, well, we have pretty similar similarities between chimpanzees. Yes, it's 55 or 60, but it's pretty close. Well, it shouldn't be close, even according to your own theory, right? I mean, what's that far ago? So you can't say that it's close when it should be far. But you know how they do it. Anything is evidence to them.
So, no? Humans and chimps are genetically more different than any two given dog breeds. Dog breeds are actually not that much more unique from one another than any two given humans are genetically. They are very different morphologically, but that's the power of artificial selection. And also, wrong again, Rawmatt, humans and chimps have precisely the difference predicted by evolutionary theory and mechanisms. So maybe double check that math.
Well, and that's the thing. Even the amount of DNA differences that separate chimpanzees from humans, even if we were to go with a conservative estimate, like 30 million, I mean, there's a massive waiting time problem. How do you fix that many differences, even according to their own model, which would suggest a split roughly maybe 6 to 10 million years ago? Because we've got roughly, conservatively 30 million differences, DNA differences that separate chimpanzees from humans. So they have a huge waiting time problem there. Well, it's a lot of speculation, and the fact that these so-called human ancestors of ours have all coexisted, intermingled, they've overlapped. It's nothing but a bushy mess. Everybody's going to see in a clip from a lecture we have later where the evidence for the coexistence of all the Australopithecines, all our so-called human ancestors. We've all lived together. There's evidence suggesting that the Homo genus ate the Australopithecines, and it's nothing but, see, the more they find out, the more confusing it gets, and they will oftentimes admit that. And another funny thing is, when it comes to Homo erectus, for example, if we were to assume, because we know evolutionists are constantly invoking massive extinction events, and they would have to, like, for example, they're doing it to account for the genetic evidence we have in the Y chromosome, mitochondrial DNA, and levels of genetic diversity, but they even have to invoke massive extinction events to account for the numbers of people on the planet today. If you look at the numbers that they're given for the dates, okay, and the dates are always all over the place, of course, can't be trusted. If you actually look at their model and take it seriously, okay, if we assume their model, let's suggest that there were roughly 1 million Homo erectus on the planet for about 1 million years, given their story. Their average time being between 20 and 30 years, okay? That would mean there should be billions of dead bodies within the time frame. Proponents of evolution suggest that Homo erectus existed. Matt, where are all the dead bodies? Because as a matter of fact, we can take all of the fossils that have been designated or that are associated with Homo erectus, and they can literally fit in the trunk of a car. So where's the billions of dead bodies? It's clear that they have never existed. It's because ape to man evolution has never happened, Matt.
Extinction events are incredibly problematic to a young Earth and aren't something that I typically touch on, but will do so here briefly. We know about the 5 major mass extinctions because they've not just left bodies in the fossil record, but also geochemical and geological traces of themselves. Because young Earth creationists require all of these events to have occurred during the global flood of Noah, the support for such massive environmental upheaval provides a kick in the crotch to the already down and out idea of a global flood. The year of the global flood has to account for massive ocean acidification, followed by a 300-foot sea level drop globally, glaciation, and the subsequent melting of these glaciers thanks to a carbon loop, followed by massive eutrophication and stratification of the seas. This would be the Ordovician extinction, and it's supported by dozens of papers detailing the geochemical and geological signals necessitating such events. Next would be the Devonian with mass eutrophication again, this time resulting in a potentially more deadly anoxia coinciding with an enormous uptick of plants in the fossil record. This was followed by another glaciation and are known as the Kellwasser and Hengenberg events. But the Permian takes the flood story and mercilessly breaks every bone in its hypothetical body. Flood geology must account for the largest detonation in the history of our planet as the Tunguska Basin exploded and the subsequent full collapse of the carbon cycle, resulting in a warming never before seen or since, and potentially hydrogen sulfide-laced hypercanes that then suffocated the land. All of this recorded geochemically and geologically. All of this in the first half of the flood year. The Permian took millions upon millions of years for the planet to recover from, and the Triassic would follow. It was less dynamic, but would kill off an enormous portion of life, with the release of CO2 causing drastic and catastrophic climate change. But the flood must also cope with the iridium layer. Iridium is only found in rich amounts in meteorites and can be found in a global band separating the dinosaur strata from the strata where no dinosaur ever appears again. Volcanism can produce small amounts of iridium, but nothing like what we see in the iridium band. Take into account as well that the Deccan traps were also exploding, and you get yet another event that geologically and geochemically precludes the flood. I've answered the lack of bodies question already, and the answer is, taphonomically speaking, fossilization is just relatively rare. This is why we still don't have that many dinosaur fossils, despite the fact that the Mesozoic was around 26 times longer than the period of time since our last common ancestors with chimpanzees. All things considered, the fossil record is pretty good for the hominins.
And it turns out the non-protein coding RNAs in our genome, they regulate nearly all aspects of the gene expression pathway. We found genome-wide functionality in the DNA elements such as ERVs, retrotransposons, ALUs, pseudogenes that they have said are nothing but genetic mistakes, produce RNAs that actually increase the expression of what? Their corresponding functional genes. ERVs are regulating gene expression in the testes, mammary glands, gastrointestinal tract. Orphan genes, go watch our 2-hour video, Destroying Gutsick Gibbon, on orphan genes. And even going to introns. We know introns are not just passive spacers anymore. They are rich in splicing factor recognition sites. We've got prediction after prediction, retrodiction after retrodiction on mutation rates, speciation rates, DNA function, but they don't want to address the data. But they want to scoff at created heterozygotes on the rate project results that suggest evidence for accelerated nuclear decay in the rocks themselves, fission tracks, radiohaloes, helium, for example. Okay, testable predictions on the flood model regarding cold slabs, for example. Catastrophic plate tectonic predictions, retrodictions. We touch on all that in that nearly 3-hour video with Jeremy. That's evidence in the rocks that accelerated nuclear decay has occurred. There's been no response, even on the field that, Gutsick Gibbon thinks destroys our model, has been addressed thoroughly. So the whole point is, what these evolutionists will do is they'll try and address 0.0001% of what we put out and ignore everything else because they know they can't address it. We're not falling for that game. Everything that's been said has been addressed thoroughly. Tonight's going to be extremely thorough as well. So I just wanted to point that out. Matt, what are your thoughts?
No, exactly. I mean, what I don't understand is you can portray something as detailed as possible, but in reality, sometimes you just say, look, this is the person that's the specialist on. This is what they have to say on the matter. And we have lots of different answers for the same problem. There isn't just one answer. So it's been answered, and it's been answered by multiple people, because you got to remember, there's conflicting models that exist even in ours, just like there is in the evolutionary model, and they compete with each other. One says, no, I've solved the heat problem. Another person says, no, I have. So there's multiple people that are working on if they even think it's an issue to begin with. I know Walt Brown doesn't have a heat problem with his model, but his is different. His model, his heat problem isn't with tectonic shifting and plate movement. His heat problem is from the actual fountains of the Great Deep Break. But that's irrelevant right now. Anyway.
Well, what's funny, I'll make one last point there, is even if there really was no solution to this so-called heat problem, and we've provided many reasonable explanations, okay? It doesn't matter.
It doesn't matter. It doesn't matter.
Because we have evidence directly in the rocks themselves. This was a prediction by the rate team before going into it. So if Erica still wants to use this heat problem argument, she's going to have a lot of work to do to actually go address the evidence directly in the rocks that accelerated nuclear decay has occurred. Because hypothetically, if there was no explanation as of yet for the heat problem, well, we have the evidence in the rocks that it occurred. We have the predictions based on the flood model. We have predictions with rapid magnetic reversals coming from Russell Humphreys, predictions on helium, predictions from Baumgartner on the cold slabs, for example. Prediction after prediction, the evidence is there. Now, there are solutions. She's going to say there is no solutions. Fine. But go address the actual evidence that is undeniable for accelerated nuclear decay. And we haven't seen that yet, and I doubt we ever will. So I would say that can be put to bed, put to rest. So
then they play a clip from Roop, co-author of this steaming pile. And Roop is not an anthropologist, nor is he a paleoanthropologist, and boy, howdy does it show. I mean this as an insult directly to Christopher Roop. His knowledge on paleoanthropology is about on par with Standing for Truth and Rawmatt's. First, Roop supposes that building tools and shelters is innately human. It's not. We see animals of all sorts doing so today. The tools, he claims, are too sophisticated, much more so than a chimp's. But what he fails to mention is how primitive they are compared to some of the later tools that we have. But that would mean no. The tools are transitional. You mean you can't just go from a heavy rock hand axe to an exquisite flint-knapped scraper? Shocking. Roop proceeds to list a bunch of hominins and how incomplete they are, but when I say he's mentioning hominins, I mean he's literally mentioning individual finds, like Lucy or Artie or the MH1 and MH2 specimens. As he did in his book, he literally does not touch dozens of specimens of these hominin species, but Most egregious is his representation of Homo habilis. He never mentions adjacent hominins like Homo rudolfensis or Homo gauntengensis, and he really shits the bed on quoting Leakey, not understanding that Leakey means the robust Australopithecines, also known as the Paranthropines, who have long been known to cohabit the Earth with the early to mid members of genus Homo.
So here's famous paleoanthropologist Richard Leakey. He comments on this finding, and he says, I see no reason that bands of Homo would not have killed and eaten robust Australopithecines when they could, just as they killed and ate antelopes and other prey animals. And indeed, they find these sites and they call them living sites. They're effectively like campsites where they would butcher remains, they would eat them, they would apparently...
Eating the missing link, how about that, eh?
Human social behavior. And so they're picture nomadic tribes that are simply hunting and eating these animals.
Crazy.
This is a stark difference from the much older gracile Australopithecines like Australopithecus afarensis or Australopithecus africanus. Robust Australopithecines versus gracile Australopithecines. Come on, how do you confuse these two? Perhaps by not having the background in paleoanthropology that would be, required or at least recommended to write a book on the subject. It's such a grave error that it makes me want to throw up, and Standing has shown this clip more than once. Next, they talk about the Laetoli footprints, seemingly not knowing the difference between something looks kind of human and something is human.
And it's just so funny watching Gutsick Gibbon with she resorts to a paper where they're trying desperately to say that, because clearly it's human, but they need to say that it's either intermediate or it's not quite modern human.
This would be like me looking at a bush dog and remarking that it looks kind of close to a dog. It is, but it's not a dog. That distinction is one that babies can make when they correctly distinguish dogs from wolves, for instance. They also mention the paper I always use to support the Laetoli footprints having been made by a non-human hominin, and then proceed not to go through it, and instead superimpose that foot picture onto the prints. So we went over this paper in the last video, which I know Standing watched, by the way, and the paper essentially concerns itself with how the animal that made the Laetoli footprints bore its weight, and how that weight bearing influenced how intermediate it was, which is to say, very, very intermediate. And Standing and Rawmatt just sit here and talk about how the prints they look human. They look human. Guys, come on. Eyeballing is not how this works. If you found this bear's hind footprints, or perhaps the hind footprints of a gorilla, you probably could differentiate them from a human's by, taking casts and looking at how the weight was borne. It's that simple.
I mean, it's funny because every single fossil, everything in the paleo community is contested. That's why what Erica will do in her response. Here's my prediction. Once again, ignore all the genetics, pick out 0.1% of it, thinking she's addressing it. Probably misrepresent it, like you've seen at the beginning here with the genetic diversity and created heterozygosity. But what she'll do is she'll cherry pick a quote from one expert in the field and then ignore all the other quotes from the other experts in the field.
Standing, can you name one individual involved in paleoanthropology who thinks the Laetoli footprints were made by modern humans? I forgot to touch on this fossil as well, which is briefly covered by Rawmatt in that it is indeed a larger Australopithecine. Rawmatt uses this to support the idea that this was actually just a human. What neither of these two mention is that the height of Big Man, the name of this, the colloquial name for this fossil, would have been about 5.5 feet tall and around 90 pounds. The prints they mention were close to the classic Laetoli prints, and in size and shape, they look the same. They're very derived. So how could we possibly have two trackways that look somewhat human of drastically different sizes attributed to the same species, that being Australopithecus afarensis? Surely nefarious secular science is afoot. No. Australopithecus afarensis was sexually dimorphic in body size. Sexual dimorphism is actually what I'm writing my master's thesis on, and many, many primates exhibit it. We have the monomorphic gibbons, where males and females are the same size, chimps and bonobos and humans, where males may be slightly larger than the females, and gorillas and orangutans, where males can be up to twice as large. Australopithecus afarensis has been considered to be dimorphic literally since its inception, so the big man, Site S tracks, could not possibly be more par for the course. My recommendation for the brain trust is to stay the hell away from Paleoanth, because you're not really doing your research. I want you to put a pin in what Rawmatt is about to say. I'll keep track of it for you as well, but keep what he's saying in the back of your mind. We're going to go through it and dissect it piece by piece a bit later with a guest.
All right, where to begin with this one? So I guess I'll just read from this quote right here real quick. Substitution rates among humans appear to be much higher rates than inferred from human-chimpanzees comparison, which are the phylogenetic methods. So when she says, or he says, these rates don't line up. They have to be assumed. They're just mathematical errors. Well, unfortunately for them, the mathematical errors are so in favor of us that they want to point out that they don't match us, but they are so far from matching their model, it's ridiculous. Now let's move on here while I break this down for you, okay? Because she specifically mentioned two separate things. One, she was mentioning, he mentioned that the models don't do the math correctly. So let's look at actually how they did the math in a couple different ones. In humans, this yields a rate of about one mutation every 300 to 600 generations, or one every 6,000 to 12,000 years, assuming a generation of 20 years. This is from a report, I would say in the late 90s, that came out, that was talking about the DNA, mtDNA and protein differences between all the great apes and their divergence rate. So this was their prediction. It would be very, 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 very slow. But that is not what we found. What we did find is this. How, and from pedigree studies, which is trio studies. Remember, her complaint was Jeanson looked at dyads and not triads, and triads are trio studies, meaning three people. So Hal was able to estimate that a mutation probability arose in the same woman born in 1861, yielding an overall divergence rate of one mutation every 25 to 40 generations. And he's talking about Parsons here. He said both of our studies came to a remarkably similar conclusion. Those studies were published back in 1996 and ran to about 1998 and 1999. But regardless, the mutation rate was exact opposite of what they actually predicted. Remember, they predicted one every 1,250 years, or one mutation every 600 to 1,200 generations. It's the exact opposite anyway. So visually what we found is the exact opposite of what they did. Evolution predicted one every 300 to 600 generations. We found one every 33 to 40. So how did Parsons actually come up with that date? Like he said, oh, if you make an error, it becomes that way. Not at all. Look, here's how he did it. Very simple. He took 133,000 years, which is when Stone King and Sherry here actually determined the last bottleneck occurred. And then he simply divided by 20, the fold difference between mutations rate that he actually obtained with Howell. Guess what? That came up to 6,500 years. So there was no error. There's no like low baseline and high baseline. He just did the math. He goes, well, if these people right here are correct and when the bottleneck occurred, and I say the mutations rate is 20% faster than expected, and I just do the math, boom. That's it. Real simple. He didn't have to do anything. These people literally believe in evolution. They're not trying to get young Earth creationist timelines. They just simply did the math and they got that as a time period. That's it.
You look good.
All for you, baby.
And now I'm very excited to talk about some of the latter fourth of this video's genetics claims, specifically that lovely Excel spreadsheet that Rawmatt plopped up there on the screen. So all of what Rawmatt just mentioned is essentially a rehashed version of the argument that appears in the book Replacing Darwin by Dr. Nathaniel Jeanson. First thing you need to know about Jeanson is that he is a creation scientist, so you may be thinking,
I don't know who that is, and I don't care to find out.
And your gut instinct is right, because, he's problematic. So Standing has this weird thing where he trusts the work of Nathaniel Jeanson, and by trusts I mean Standing seems to worship the ground that Nathaniel Jeanson walks on, and also seemingly wigs out of his gourd whenever someone disparages the man. So can we trust the work of Nathaniel Jeanson? Well, considering that the nickname for bad faith acting in this video is Jeansoning, you might know my thoughts already. Nathaniel Jeanson is a part-time Answers in Genesis employee and holder of a PhD from Harvard in cell and developmental biology. Standing likes him because Jeanson claims to have found that the human mutation rate places mitochondrial Eve as the real biblical Eve from 6,000 years ago and Y-chromosomal Adam at Noah some 4,400 years ago. He also likes him because Jeanson supposedly makes predictions. To Standing, it doesn't really matter that Jeanson isn't testing many, if any, of these himself, but Standing is still frequently quelled by his buzzwords. Now, I am of the personal opinion that Nathaniel Jeanson is a bad faith actor. I don't know this for certain, and it's not necessarily the position of the guests that show up here today. In fact, I want to make it very clear that I don't speak for them. I'm going to let them speak for themselves. With that out of the way, let's go ahead and welcome our first guest here to discuss Jeanson's wild mutation rate and Standing for Truth's use of HapMap data to attempt to justify young Earth creationism. This is Crispr.
I found myself for quite a while working as a lab manager in a cancer lab before deciding to go back for my PhD in evolutionary biology, which was what sort of always interested me the most.
Right.
And so I've got, it's like 7 years of experience with like actual like hardcore molecular type sciences in the actual cellular and molecular side of things. And now I'm doing evolutionary biology, and that gives me a breadth of experience that I think most people don't have.
That seems like a unique range.
Yeah, it's nice. I feel like, because, so I'll tell you that, like, Neil Shubin does what I want to do, right? He's right at that cusp of, like, of the real cell and molecular manipulation combined with serious morphological and evolutionary study, and that combination of features is what I'm really interested in.
With credentials out of the way, we decided to jump right into HapMap data, which is what Standing for Truth uses to essentially buck the idea of the out-of-Africa hypothesis in favor of this out-of-left-field proposal by him and mostly other creationists that humankind began and indeed migrated out of the Middle East.
So I checked out the HapMap video, and before I watched it, I did what Standing recommends, and I watched Carter's lecture.
Okay.
I watched Carter's lecture once, because it's not complicated, right?
No, right. It doesn't get too in-depth.
And so, after that, I listened to SFT talk about it, and SFT is going off about, the evolutionist model, they have to invoke all of these bottlenecks. You can't explain all these bottlenecks that evolutionists have to invoke. Now, firstly, I don't know how many times I've told him this, but the Tobo bottleneck is not broadly accepted by the scientific consensus. Generally speaking, most people don't think there's any real evidence of a 70,000-year bottleneck in human populations or in the populations of any of the other great apes in Africa.
Right.
It's just like you do see it in Sumatran in the Sumatra populations because they were there, right?
Yeah. Well, they were genuinely isolated.
Well, but also they were closer to the volcano.
Right.
So, but not in the human population. There's some suggestion of it. I'm not saying that it's wrong. I'm just saying that it's not required to explain the diversity that we see. But the funny thing about this is there was a there is pretty strong evidence of a bottleneck right around the origination of anatomically modern humans, about 250-ish thousand years ago.
Give or take, right?
But that's actually consistent with speciation occurring, so like that's not surprising necessarily. But that's not the one he talks about. He always talks about the 70,000 years ago one, which is the Tobo eruption.
Right.
Anyway, Standing also talks about the fact I had to watch this Carter thing. I knew I had to watch it like 5 or 6 times. So I've watched it so many times now to understand it. And it's like, okay, you watched it that many times, and you didn't pick up on Carter saying that he got his results by invoking like 5 or 6 sequential bottlenecks in Noah's family tree, for which there's not even biblical evidence. And support.
Really?
Yeah, that's how he gets those numbers.
Oh my god.
So here I'm going to run a model. And here's my 3 numbers. I'm assuming people mature at age 50. That a female goes through menopause at age 350, and there's 20 years of spacing between each child. Now, that sounds kind of ridiculous, doesn't it? But I do that because I can't have more children than that per generation, because at 800 years, my computer runs out of memory with 100,000 people. You can do amazing things with computer if you have the horsepower. Well, HP laptop, I mean, it's a pretty modern laptop, but it couldn't handle more than 100,000 people. Fair enough. So here's my results. Remember before I had Adam and Eve, black and green? Well, here I'm replacing that black and green because I have to do multiples of these with just a straight red line. Adam and Eve, 0.5 every single allele. The population 100 years later looks like this. Not too surprising. 100 years after that, the population looks like this. 100 years after that, the population looks like this. 100 years after that, 100 years after that, 100 years after that. Where's this going to stop? There. Look what happened. In my exponentially growing population, genetic drift stopped once the population got above a couple of hundred people. Because once it got above a couple of hundred people, that next generation is going to be a little bit larger. Well, they're grabbing almost all of the genetic variation the previous generation and popping it into the next generation. That means the current world population today, how many people are in the world? 6 billion. Genetic drift isn't operating. Or if it is, it's so slow, and even in geological time, it's not going to occur. I mean, mathematically, it's possible, but it's going to be really slow. But this is only part of the biblical model. I'm only slightly there, and I say, wait a minute, I can start with Adam and Eve and get this, but I'm not always as far as I want to be. So, well, what's next in the biblical model? It's the model of the flood. What happens at the flood? 3 full sibling brothers and their wives. I don't know how closely related the wives are, so I'm going to pull 3 random females out of the population, giving evolution the benefit of the doubt. 3 full sibling males, 3 random females from the population, they form a new population. That's called inbreeding. What does inbreeding do? It leads to genetic drift. So here's my model again. Same exact parameters as before. Same exact parameters. There's Adam and Eve. But this time, 100 years later, 500 years after Adam and Eve, I'm going to throw in a bottleneck. Now, I didn't wait till 800 years. I wanted to do 1,500 years, but I couldn't. My computer couldn't handle it. So I did it at 500 years before my computer locked up. And I erased all the data, and I could start building a population again. So at 500 years, I'm reasonably confident genetic drift is not going to occur anymore. I think that's okay assumption. Boom, genetic bottleneck. 500 years after the genetic bottleneck. Let's do it again. Boom. 500 years after that, it looks like this. Let's do it one more time. Boom. What are you seeing? You're seeing the HapMap data. Now, I'm assuming exponential population growth, which might be true, but I'm also assuming random mating, which is ridiculous. Random mating doesn't happen in any world population, or we would all be middle brown color with brown eyes. And we're not. Humans do not randomly mate, and we have not randomly mated throughout all of human history. Period. But also, is it fair to model multiple population bottlenecks when the Bible only talks about one, Noah's flood? Actually, yes, it is. Because it doesn't matter what happens to the world population. It only matters what happens to to Noah's family. It's Noah's ancestry that counts. Nobody else counts. And if you think about people in the pre-flood world, they're marrying, they're having children, they're having children, they're having children. If those people spread out, they're going to be lost to one another. There's not going to be any random mating. If there's any, and there's lots of violence in the world. Well, if we have any kind of violence, we might get clannishness. We might get family snobbery. Might get geographic separation. Kings forming and kings demanding that they marry only some people, excluding others. Whatever. If there's any degree of non-random mating in Noah's lineage, this occurs to some degree. Now, the reason I'm throwing out multiples is because I don't know to what degree, but I do now know that it is possible, and that excited me tremendously. But look at something else very interesting. See this down here and up here? These are alleles that have gone to fixity. Some of Adam's genetic diversity might have been lost even prior to the flood. I had never considered that before. Some of the genetic data...
So he's standing complaining that we're invoking bottlenecks that we're not even invoking.
And then he turns around and supports someone who's invoking 5 arbitrary bottlenecks. Okay,
right.
Awesome.
Yeah,
awesome. Cool.
Now, the other thing is that Carter's entire point with the HapMap data, right?
Yes.
Ignores the concept of subpopulations, because the real thing that people point out when it comes to diversity numbers is not the level of diversity itself. It is the distribution of diversity. That's the killer.
Right.
Right? It's not just here's how many differences there are. It's the fact that the differences that exist are distributed in the way that they are. And it's not just Carter likes to point out, well, if you sort up like all of these SNPs by their what percent they are in the population, the allelic frequency of each SNP, oh, you get a nice straight line. Okay, fine. That doesn't matter, right? The interesting thing is that you have one SNP that is 70% in population X and 10% in population Y and 0% in population Z, right? And then some other SNPs completely different, some other SNPs completely different, right? And
so you're not getting an accurate representation.
Right, and all of this lines up with the concept of diverging subpopulations that cannot be made to be consistent with the biblical model.
Yeah.
So it's not the diversity itself; it's the distribution of diversity that matters. And now this is something. When you look at the 1,000 Genomes Project, and especially, I'd have to pull up the paper on it. I was looking at it just the other day, but like it really gets into like the full sequencing of like 2,500 people from all of these different populations around the world. And you just see like how many differences there are and how divergent they are population by population. You cannot align this with a young Earth, any kind of young Earth model that doesn't invoke magic. I don't know why they don't invoke magic, by the way. That would be simple. You've got God on your side. Just say it's magic and be done with it.
So Standing's model, and thus Carter's model, fails essentially because it fails to incorporate subpopulations, their distribution, and additionally adds 5 or more arbitrary bottlenecks that are seen neither in the scripture, but more importantly, in the genetics.
Oh, actually, so here's another thing about the HapMap data that just occurred to me, which is that Carter, in his presentation, he talks about how they use Mendel's accountant, which I should circle back to in a minute. But he uses Mendel's accountant to show here's the distribution of mutations that we would expect in a population over some amount of time, so you see that there's mostly very low frequency of alleles. And so obviously, again, this is ignoring the concept of subpopulation divisions. But the funny thing about this is that the way that he gets a straight line, the way that he replicates that straight line, is he has to invoke additional mutations, right? And so he's going to say mutations over time are what's going to like help straighten out the edges of this line. Like, but why don't they straighten out the line at all in the model that doesn't start with Noah? That's
Right.
not mentioned.
Oh my gosh. So with the HapMap data out of the way, let's move on to Nathaniel Jeanson.
They're starting with Parsons, right? And HAL 96. And all of these old papers, they got a particular result. And if you look at HAL, once he upped his sample numbers, he got a much lower number. He got a much slower rate. And Parsons also got a much slower rate when he upped his numbers, even though every time I read Parsons, it gets worse, because I went from thinking that Parsons was a forensic scientist operating outside of his depth to thinking subsequent publications are blatantly dishonest.
Really?
Yeah, because I'll tell you why. So his original paper, the Parsons 97, he has 4 populations: the AFDIL group, the Oxford family, the Amish family, and there was some other hereditary disease pedigree that he was working with. And the hereditary disease in the Amish family got much lower rates than what he reported. And the Amish family got like dead on what he reported, but with only 32 meiosis, right? And the AFDIL got like twice the rate that he reported. But he averaged them all together, and it came out to be, this 2.76 or whatever the number is. And then in his second paper, not a second paper, he writes a response to Jeanson et al. And basically says, well, look, I've increased my numbers and I've gotten an even faster rate. But what he does is he compares. He did his original 97 AFDIL. I'm doing this all from memory, by the way. This is how many times SFT has made me read this goddamn paper. Doing this from memory. Okay, so the 97 reported on 327 generations, 10 mutations. That is all 4 groups. The AFDIL was 125, I think, something like that. Generations with 7 of the mutations, and there was 1 each for 32, 80, and 90 generations. And if you want to know the actual number from the AFDIL, it is 32, 80, and 90, I believe, 90 something. So do the math on that, and you'll figure out the remainder from 327 is whatever the AFDIL is. Anyway, in the 98 response to Jeanson et al., he has doubled the generation count for the AFDIL, and he has found an additional 3 mutations. For 300, he now has 306 generations of AFDIL, right? And he found 7 in the first half and 3 in the second half. Well, that's very different from one another, isn't it?
Hugely so. Hugely so.
But here's the thing. He, instead of comparing, and then what he does is he compares the 2.76 number that he gets from the 1 out of 32 from the Oxford family to the 10 out of 306 in the AFDIL, which is like 2.97 versus for the 98 versus 2.76 for the 97, when the original AFDIL number is like 5.9.
So what's his justification?
He likes those numbers, I think, is my impression, because like you cannot combine when you have numbers from 4 different groups, because these are 4 fairly different data types, because the AFDIL is like recovered DNA from forensic samples. So this is, they found tissues out in the field of recovered bodies from unidentified service members that they're comparing against family members that they're trying to match up with, right?
Right.
So that's one group. Now, by the way, in that type of sample, you're expecting some degree of sample degradation and possibly some degree of,
contamination.
more importantly, some degree of contamination.
Right.
Because if you have, 3 or 4 guys in a foxhole and a grenade lands in there,
yeah, you're stirring them up.
yeah, it can get kind of nasty.
Yeah.
So specifically, and the harder it is to identify these remains, the more likely it is that you have potential contamination. And especially when they're talking about the fact that they have these really, they're not particularly sensitive to the possibility of heteroplasmy and all this other stuff. And I don't know what to make of this. But then you compare it to the Oxford data, right? And not the Oxford data. The Oxford families there's only 32 generations, right? So it's a low sample size. And so you have 1 in 32. Maybe that's just the chance that you got one fast. Let's, see 2 or 3 times the number of samples. And I do think you can reasonably combine the Oxford data with the Amish family data and the other hereditary disease data. Well, now you have 3 mutations in 160, 180, or however many generations it is, and that's actually fairly in line with a lot of the other reports. Of a comparative number, I was saying 2.7, 2.9. It's 0.9 something for those, and that's consistent, because the Howell 2003 is the meta-analysis is 0.95, which is still faster than the phylogenetic rate. But here's the thing that Jeanson is really dishonest about, is that you'll notice the papers that he cites, they stop 5, because 2008 you get the Santos paper, which basically conclusively demonstrates exactly what's happening and confirms the prior explanations for the discrepancies in the rate, which are basically a variety of just biological processes relating to both the heteroplasmy and just the nature of reproduction, coalescence, and all these other things. I use the analogy of if you have a sports car that can drive 120 miles an hour, does that mean you can get 10 miles across town in 5 minutes? No, it doesn't mean that, because you have to deal with traffic.
There's a whole host of additional variables.
You have to deal with terrain. You have to deal with the roads, and you cannot name a single thing that affects the timing that doesn't slow you down. Everything you can possibly name necessarily slows you down from whatever that maximum rate is. And what they're finding is a maximum rate. And so, yeah, when you go out over a longer period of time, the number gets lower. And we know why, because Santos demonstrated it. But then Jeanson starts citing these papers from 2014, 2015, 2016, when the next generation sequencing data starts coming out. And he doesn't bother to mention the fact that when they get results consistent with the 2003 to 5 numbers, like the HAL 2003, Sigurd their daughter, all of these other papers, well, of course they're consistent. Why wouldn't they be consistent? We expect them to be consistent because Santo's 2008 paper already explained it. So it's just bad.
He's choosing essentially to lie by omission and leave out the explanatory factors that are at play in getting the rates that he's getting.
Yeah, and the thing is, I've almost gotten standing to say it out loud. I just want to ask him, it's like, okay, do you accept the fact that whatever we say the rate is from one generation to another, do you accept the fact that rate must be faster than the rate from one generation to 100 down the line? It must be faster.
Yeah, by definition of how reproduction works.
It's just biology.
Right.
You can't. Biology and math combine to say this is a maximum speed. Actual speed over time must be lower, and it must go asymptotically from one to the next. It's a sigmoidal distribution. There's an inflection point at somewhere in there. I don't know exactly how many generations it is, but somewhere in that line, there's an inflection point, and you go from basically the one generation to the next rate to basically the phylogenetic rate. We have mathematical explanations for this, which, by the way, I have actually programmatically simulated. And all of these numbers, even if you use the simplest possible math, all of these numbers, except for Parsons, will get you to at least like 20,000 years.
Right.
So,
and Parsons was the one that was inherently flawed to begin with, due to essentially his sampling and his methodology.
Yeah, right. And and Parsons, if you actually look at the if you look at the confidence interval on Parsons, it's something like 900 years for the most recent common ancestor of all humans. It's something like 900 years to 1.9 million years.
So they're genuinely just.
That is a big confidence interval.
Oh my god. I cannot believe that. So let me see if I'm getting this wholly correct. Because usually when I focus on what I think Jeanson's general problem is, I actually haven't looked at where he's pulling his necessarily where he's pulling his mutation rates from. I've always just kind of been like, okay, well, I'm sure that there are ways to get certain mutation rates, particularly in papers that old. But so let me get this straight. So Parsons initially, when they came out, Parsons and collaborators, they come up with this super fast mutation rate because of a poor methodology as well as bad sampling. After that, subsequent papers that were also getting relatively fast rates were eventually superseded by Santos et al., who came out with work that was like, yes, we are getting fast mutation rates. This is the reason why the rates are somewhat faster than they would be.
Right, and I want to be 100% clear on this. The pedigree rate is absolutely faster than the phylogenetic rate.
Right,
no
by
question.
necessity. By necessity.
It is faster by about maybe five times or so, right? Give or take, depending on exactly which paper you're looking at. Maybe as much as 5, let's say 5 to 10. Parsons got 20. That's wrong. Okay, so it's not just that the idea that the pedigree rate is faster is wrong. What we're saying is, first, Parsons is wrong entirely. Second, we know why the rest of them are faster than the phylogenetic. Here is the reason why. Look at Santos 2008. Like, this is, so, yeah, it's two separate things. First, yes, the pedigrees are faster, not as fast as Parsons says.
Right. And then, after all of this, Jeanson comes out using work from 2013, 2014, he's taken their fast mutation rate, but omitting the reasoning for said mutation rate, and is using it to validate his hypothesis of a 6,000-year-old Earth and progenitor of humanity.
Yes, which he's mostly using papers that don't eat. And here's the other equivocation that happens, and it's such a quick sleight of hand that I think it's often missed. Is that what he does is he says Parsons got a rate that agreed with 6,500 years. Other authors got a rate that agreed with Parsons, which is false.
Okay.
And then later authors with next generation sequencing got rates that agreed with this third group of authors with NGS got rates that agreed with the second group of authors, which is true, right? And so there's this equivocation that happens because what he's trying to do is he's trying to say that all of these authors, both the original like Sanger type sequencing, pedigree sequencing that was done from 98 to 2003 or so, and the later next generation sequencing that was done in the mid 2010s, 20-teens, that they all agree with each other. And that's true, they do all agree with each other, except for Parsons. Parsons is the odd one out. The rest of them all agree with like 30,000 years or so, using the simple math that is, by the way, wrong.
Right, yeah. Even getting to 30,000 years, that's your best case scenario that is also incorrect. Like, you're not even getting to that 6,000 that is required for the young Earth.
Right, and then using the simplest possible simulation that I could think of, which is by definition the most generous to Jeanson, I got about 1/3 the rate. So you get your 30,000, then goes up to about 90,000, and that's with just the most simple possible reproductive biology, without considering rate heterogeneity, without considering a lot of other things. Just like you're going to lose some lineages each generation. Just that alone gets you to about 1/3 the rate. So now you're at least at 90,000 years if you look at everything but Parsons. And that's before rate heterogeneity, which is the thing that really kills them.
Oh, wait, you're serious? Let me laugh even harder.
So just to be abundantly clear, Nathaniel Jeanson's work with mutation rates is so ridiculously flawed, and we're not even done yet. Remember that Excel spreadsheet, that table that Rawmatt put up at the end of the video? You know, the one that he cribbed from Jeanson? Let's look at that.
Yeah, I think I had. Yeah, so this is always fun. So the first thing is 15 years. That's insane.
But it gets so much worse than that. Let's use an example to see exactly how Jeanson's methodology hashes out when compared to what it is that we actually see. Imagine that we have a sequence that's approximately 10,000 base pairs long. The mutation rate for 9,999 of these sites is 1 × 10⁻⁵ mutations per base pair per generation. The mutation rate for the one remaining site is 1 × 10⁻⁻1 mutations per base pair per generation. Before you ask, as I know you're just dying to get ahead of the gun here, yes, mutation rates do vary depending on the site it is that we're looking at, and yes, this is well supported by the literature. Now Jeanson comes along, eyeballs the situation, and decides to apply his methods to it. He says that the population of humans originated 200,000 years ago according to evolutionists, which he calculates should be about 10,000 generations ago. So he calculates that there should be 2,000 differences in the sequence between the two most divergent individuals if they really did diverge 200,000 years ago. But Jeanson shows that we only find 101 differences between the sequences. At this point, he would conclude that there simply must be a problem with evolutionary timescales. He takes it a step further, though, and proposes that not only do the evolutionary timescales not make the cut, but the young Earth creationist methods do. So, what's going on here? As it turns out, the problem is that Jeanson's analysis doesn't actually consider which sites the differences are in. In this example of the 101 observed differences, there's only one difference in site X, and the other 100 differences are spread throughout the rest of the sequence. This would precisely match evolutionary predictions of how many differences should be present when taking into account the relative mutation rates. Over the 10,000 generations, 2,000 mutations did indeed happen, but 1,900 of them occurred in a single site because of its higher mutation rate, that being site X. Since it's only one site, it would show up as a single difference in comparison with another sequence, no matter how many mutations it actually experienced throughout history. The real question is, why didn't Jeanson know that? In all seriousness, after something like this happens, I just don't know how Jeanson could possibly respond, really. Say the line, Jeanson! Created heterozygosity.
Well, so one thing I actually did learn from Carter's presentation, and yeah, I'll be honest enough to say that I did learn at least one thing from that presentation, is that Noah's firstborn was born when Noah was 400 years old, and that's kind of problematic for them, because I think that Noah's wife was also pretty old at the time.
Oh, and, superbly.
And so that's going to really screw with their generation time numbers, because you're losing like 10 generations right there, 15, or more, because they have to use the 15 number to get up to the 120 that's actually observed, but they can't because Noah. So that's pretty bad.
Yeah, they end up losing a player in the game with that they kind of take for granted in all of the models that I've seen.
That's his starting point, at least. Also, something to watch out for is whenever Rawmatt talks about it, he always likes to cite Ann Gibbons as a separate study. You'll always hear him use the word study. Like, Ann Gibbons had a study. Like, that's not a study. That's a popular science level write-up.
Yeah, a pop sci article. We love those. I also get from them a lot too. They're like, well, the FBI uses the Parsons paper.
Yeah.
That's
I'm partial to my analogy there, is that Parsons has he's figured out a real great way to make scrambled eggs, but that's not helpful if you're trying to make an omelet.
Yeah.
Right? It's just
absolutely. It works in the short term or I guess worked past tense, because I'm sure that's kind of advanced since then, right?
No, there's nothing wrong with it. Like, what the FBI has accepted from Parsons is that for forensic samples, you're more likely to get to detect a difference, right?
Because it's a single pedigree,
because
right?
You're more right, yeah, and because you're more likely to detect a difference than people would have thought. And so now what they say is, if you get one difference, you call that inconclusive. If you get two, you say it's not a match. If you get 0, you say it's a match. If you get 1, it's inconclusive.
Groundbreaking.
Yeah, which is, I think, perfectly valid. There's nothing wrong with that.
That's
if Parsons was limiting his work to his field of forensic science, I wouldn't have a problem with it.
Yeah, he wanted to dip his toes into, like, paleogenomics and the application in human population genetics through time, and that's when it becomes problematic. Yeah,
and you get all of these... Oh, actually, so there's one great thing that I want to make sure isn't missed, too, is that Matt especially, he likes to say that, the problem with some of the Studial or Jeanson or I think Boone was one that got rates of 0. They found no mutations in some, like, 100, 200, 300 generations. Is that these are all low diversity populations, which is funny because why would it matter how diverse the population is if you're looking at the actual rate of observed mutations in a pedigree?
Right, the population has absolutely nothing to do with that.
Because, like, now you're saying that, like, does it matter if I'm a different race from my if I'm a different race from my neighbor? Does that change the mutation rate in her ovaries? No.
Oh my gosh. This harkens back to I'm pretty sure I saw you on the thread over at Peaceful Science, but on Jeanson's predictions using the Y chromosome to try and, like, guesstimate past populations. Like, it's literally just, that's not how any of this works. Like, you cannot use Y chromosomal mutation rates and back that up to get a general population, because it's always necessarily going to be growing from the original progenitor, that original male.
Yeah, I mean, well, it's just people. So I hear a lot of people say, it's like, oh, you think that all of this came from a population of 1 or 2? No, there are lots of people at the same time. This is coalescence. I can draw this out for you. It's not. I think I actually, like, typed it in. It's like, you say A B C D A B C A B A, right? And you look at that in a nice little row, and you see, oh, well, that makes sense. There's the same consistent population numbers all the way down, but if you have an average number of children per couple of 2, so this is replacement, so you have a constant population size, and it's 50-50 whether or not they have a girl for each kid. So that means that if you have 4 couples, that's 4 women. You're going to have 4 girls on average, and one couple is going to have 2 girls. 2 couples are going to have 1 girl apiece, and the 4th couple is going to have no girls. So what happens? Like, it takes about 3 generations for you to have only one lineage.
Golly. Oh man, yeah. All of this is stuff that I genuinely had not considered. The breadth of how many different kind of supports from the general Jeanson lattice are so flawed is insane to me.
Ouch! I can't imagine that that's going to be easy to come back on, unless, of course, you simply don't engage the criticisms. Here's the part where Standing in the Gang will vehemently double down on the work of Nathaniel Jeanson, suggesting that if we're so sure that his work is bad, why don't we just confront him then?
Except people have. Jeanson will seemingly engage no one outside of the Answers in Genesis outlets, running from Facebook arguments and ignoring the emails of dozens of different people who present questions and skepticism. The real question is, if Jeanson's technical work on these mutation rates, found exclusively in the AIG archives and in his book Replacing Darwin and nowhere in the general literature, is indeed overturning the entirety of evolutionary theory, why not just publish it in a legitimate journal? Why hide it away in an echo chamber and then flinch away from the majority of criticism? Maybe. Okay, this is going to be a little bit crazy, but bear with me. Maybe it's because Jeanson's work is bad. But it's worse than that, really, because Jeanson also ignores or hand waves away all the data that actively precludes young Earth creationism. He entirely dismisses radiometric dating using many of the same complaints that we covered in this very video, and doesn't touch the heat problem at all. Kinda like someone else we know. But there's a lot of very interesting genetics work that also precludes his hypothesis of a 6,000-year-old Earth. And for that, I'm pleased to introduce you to our second guest of the video, Dr. Joshua Swamidass. Dr. Joshua Swamidass, tell us about yourself, your background, your work, and kind of what we're going to be talking about here today.
Yeah, thanks for having me, Erica, or Gutsick Gibbon. I still don't know quite what that means.
Hey, I tried to explain it to you on the forum. It's a cartoon thing that I made up. It's not as nearly as interesting a story as people tend to think when they ask that question.
All right. Well, so you even mentioned the forum. We're talking about the Peaceful Science Forum right now, right?
Yes, we are, which is included in the description for anybody who wants to peruse the site for some very interesting conversations.
Yeah, so we're a group of scientists. I'm the founder of this group. There's a group of scientists of all different sorts. Some of us have religious backgrounds and beliefs, and some of us don't, that are trying to just do a good job engaging the public. And we have a forum where we take questions and a blog, and we're doing other stuff too. And one of the things too is here engaging with you. So I'm a scientist. That's what I do with most of my time. That's what I get paid to do. I have grants from the government, doing a lot of work in applying artificial intelligence to problems in biology, chemistry, and medicine. I'm a computational biologist. So I'm also a medical doctor too. So I got both degrees in graduate school. So I was in school for a long time. And then also, I've been doing this stuff, trying to advance science and engage the public with science through peaceful science, which has been really fun over the last few years. Now, I was raised young Earth creationist, just like some of your friends.
Yeah. We love our young Earth creationist friends. We do.
I do, actually. A lot of my family still is young Earth creationist. And
wow.
There's just a point as I learned more about biology, it just didn't make as much sense to me.
Right.
And then, to be honest, as I read Scripture more closely too, I realized that the problem wasn't with Scripture. It was really what they were telling me Scripture said, but it didn't actually say. But that's a different issue. This question about Adam and Eve, though, has been something that I've thought is really interesting for a lot of reasons. Part of it is that I think that it's not a question that the scientific community has really engaged in depth for the most part, and there's been a tendency to even make straw man arguments of Adam and Eve, which I don't think is really the best of science. I think we want to kind of see what could work. Maybe there are ways that it could, maybe there are ways that it couldn't. That's part of the reason why I ended up writing this book too, The Genealogical Adam and Eve, which really surprised a lot of people. Where I showed that, Adam and Eve could have been specially created even as recent as 6,000 years ago, ancestors of all of us. There'd just be people outside the garden. And that's something that even as a young Earth creationist, I used to wonder about people outside the garden when I thought about, Cain's wife and the Nephilim and all that sort of stuff. And it turns out there's a long tradition of that. And if you're open to the idea of people outside the garden, well, then all of a sudden there really is no conflict. And that was surprising for people. I think a lot of people were really surprised by that, but it ended up actually being really good science and getting a lot of people's attention. And you might be wondering, wait a minute, we would all descend from them just 6,000 years ago? Well, it has to do with this difference between genetic and genealogical ancestry, but that's a conversation for another time.
Yes.
Today, we were going to talk about some of the different young Earth creationist models out there for Adam and Eve, and where we think they stand with the evidence, and some of the evidence that we put together to help test that.
Right, and that's the important thing, that we have to appreciate when we're looking at discourse, right, in the scientific community. The idea is that if you've got a good idea and you present it, if the data is good, it's going to be considered. That's just how science is meant to work.
Yeah, that's actually something that I think my book actually really demonstrates. It was surprising to a lot of people. So a lot of people who weren't as familiar with science thought, well, wait a minute, you're proposing the idea of a de novo created Adam and Eve. All these scientists are going to think it's ridiculous. And, yeah, sure, I don't think, atheists believes that Adam and Eve were de novo created, but that's not exactly what I'm arguing. What I'm saying is, let's say that they were. That's the hypothesis. Is there any evidence against that? And how do we look at that? And then they looked at that analysis, and it was good science, and they accepted it. I mean, atheists even endorsed the book. And that was really surprising to a lot of people. But the fact of the matter is that science isn't really ideologically driven that way. I mean, certainly scientists are people too. We have preconceptions and bias, but we can also overcome those things too if we have good science behind it.
Yeah, absolutely. It's good science, and it's good science for the individual, right? And it's a community that is willing to look at every possibility, so long as the data is there to either support it or potentially not support it, right?
Yeah, I'd say the core thing in science is that we have to be honest with the data and we have to be rigorous with it. So
absolutely.
Rigor means that we don't just look at a narrow portion of the data that makes sense to us and ignore everything that doesn't.
Yeah, absolutely.
That's not what we do.
Yeah,
And honest means that we don't misrepresent it. And a big part of that is clarity. So one way how things can get can really struggle with honesty is when we're not clear about what we mean about different things, and we kind of start equivocating and using things mentioned out. So I think one of the key things you get trained to do as a scientist is slow down, really clearly specify models, and really clearly show how each line of evidence really relates to it.
Yeah, a standardization of a methodology, clarification of terms, all these kinds of things are really important so that not only you can be properly peer reviewed by colleagues, but so that this can be accurately represented to the public if and when it does reach them. So tell us about your work, the most recent 4 alleles.
Yeah, so what you're getting at is we did some work to look at when the most recent 4 allelic lineages arise, or time to most recent 4 alleles that you can see when you look at human genetic data. To just jump ahead, it turns out that that the time that you compute is around 500,000 years ago. And for a lot of different ways of understanding Adam and Eve, and we'll talk about that in a moment. What I want to do is list out the different ways that you can think about Adam and Eve, or
absolutely.
Are. But if you're going to say that we all of us got all of our genetic information from Adam and Eve by direct descent from them, that means that Adam and Eve would have to be, if they had biology similar to us, they would have to be 500,000 years ago in the past or more ancient. And that works for some Christians. That's the view that some Catholics take, and William Lane Craig, who I've been working with too, he's kind of going down that path, and that makes sense to him. And the evidence just doesn't tell us either way really how big the human population was back then.
Right.
And that's okay. I mean, and that deviates though pretty strongly from young Earth creationism. The way how it's typically presented. Now, I don't want to say that it proves young Earth creationism wrong. I think what I would say is that that it leaves a really big question for them. How do they make sense of that data? And they haven't really responded to that yet. And it does seem to rule out a lot of different models that they proposed. And maybe there's other models that make sense, but most of them don't seem to make sense.
Right. So if you would, for our audience, for maybe some laymen out there, could you explain to us why it would be difficult to take current genetic data and extrapolate backwards into populations of the past?
Yeah, so to extrapolate, to infer that, well, it's just a lot that we don't see. It turns out we do get a lot of really powerful, important, and interesting information about our past through DNA, but it just doesn't tell us everything. We tend to see it as if it was kind of like the Eye of Sauron from, you can just look anywhere with like godlike ability or nearly godlike ability. Yeah, it's a lot more like a streetlight in Manhattan. You can learn a lot about. There's a lot you can learn about, from a streetlight, or maybe in like a series of streetlights. But it's just a streetlight. There's a lot of stuff that's outside. There's a story that a lot of scientists like to tell each other. I first heard it when I was an undergrad, from George Chandy, who was a biologist at University of California, Irvine, in whose group I was studying. And he said, and this isn't his story, I mean, people said it for like about 100 years, I think, in science. It's science is like a streetlight. There's like this story about this this police officer comes upon a man kind of searching through all of the bushes underneath the streetlight trying to find his keys. And the police officer gets in there and starts helping him, trying to help him find it, and says, okay, so you lost your keys here and you're trying to find it. And the guy says, well, no, I lost it over there. And he points down the street. And he's like, well, wait a minute, why are you looking here? He said, because this is where the street light is. There's no light over there, so I'm going to look here where the light is.
Right, yeah. That's such a perfect way of putting it.
Yeah, and so the thing about it is we think, it's right, science gives us light. Genetic information gives us light. But it doesn't give us the light that we need to answer all the questions
right.
we dare to ask.
It's spotty, and it can help us. It's like looking at a jigsaw puzzle that's 40% complete. You might start to get an idea of the complete picture, but until more information comes to light, it's a suggestion.
Well, I'd say it's even worse than that. As you go back in time, it's blurrier and blurrier and blurrier.
Right.
And we can have our preconceptions of where things are, but that doesn't mean there's hard evidence.
Right. The puzzle starts to be it goes from a 24-piece puzzle with half the pieces missing to a 1,000-piece puzzle where you've only got 10% of it complete.
Yeah, exactly. And the way I put it in the book I wrote is that science is a little bit like genetics, I would say, is a little bit like a streetlight, where it gives you a very good view of our very close genealogical ancestry and relationships. And it's a little bit like a telescope too, in that it can tell us a lot about population histories in the distant past. Like there's very good evidence that we share common ancestry with the great apes. And you can see that in our genomes. But it has real blind spots. It has if you're looking at a telescope, if I'm looking at a telescope at something way, way down there, I might see something, miles and miles away, but I wouldn't be able to see my wife sitting right next to me.
Right. Yeah.
And so that's kind of how it works, where it doesn't actually give us nearly as detailed view as we might think about the past. And so if you get back about 500,000 years ago or so, it's very hard to know much about, an Adam and Eve back then.
Humans, like nearly all other mammals, are diploid animals. This means we have two alleles at each genetic locus, one inherited from each parent. Using known rates, we can track backwards allelic diversity and generate a time for when the last most recent 4 alleles shared by all living humans existed. Dr. Swamidass has calculated this to be around 500,000 years. This, of course, falls in line with evolutionary theory and conventional timelines for the age of the Earth. So how do creationists deal with the concept of the most recent 4 alleles? It turns out there are around 4 models.
Let's talk about a couple of these young Earth creationist models.
Yes, absolutely.
So we'll talk about a few of them. The first one we'll talk about is a straw man of their position. And I think it's been really, really used in a way that doesn't make a lot of sense. The idea there is that Adam and Eve are homozygous clones. They each have 2 genomes, like 2 copies of a genome, right? So there's 4 copies, but they're all the same. They're all copies of the same thing. They're all identical down to the base pair.
Cookie cutter, yeah.
Yeah, and they've been howling for years that that's a total straw man of their position, and I agree. That is probably the most implausible understanding of Adam and Eve possible. Okay. And so that means that all genetic diversity that we see today has to arise somehow by natural processes afterwards from them. Okay, so the idea there is that they're homozygous clones, which, to be clear, is not like humans today. We don't see anyone today that is a homozygous person on every site. We don't see anyone alive today that's a homozygous clone of another person. Like, so this is a completely bizarre view of what, I mean, they would not be humans like us.
Right.
And then, then by natural processes, things kind of diverge from there. And that's where we get all the diversity, in the world. And of course, there's the Noahic bottleneck with Noah, with him, his 3 children, his wife, and their 3 wives. Which we'll get to in a moment, but that's there too. But then from there, it's just natural processes. So that just turns out to be very inconsistent with the data. And we'll see that really quickly when we look at the time to most recent single allele in a moment, too, which we could talk about. But for the time to most recent 4 alleles, that means that they wouldn't even have 4 alleles. They'd only really have 1 allele, or one version, one flavor of DNA for each position in their gene.
Right, just repeated multiple times.
Yeah, and so that's a problem, right? So that's the first model.
Model 1, homozygous clones, is indeed a straw man, so it's not really relevant, but it's still worth mentioning as it doesn't actually work. The homozygous clones simply cannot generate enough diversity for what we see in the modern human population, particularly when up against the explanatory power of the conventional science model.
The second model that's been important is looking at them and saying, okay, maybe they weren't clones and they were heterozygous, meaning that they had different flavors of DNA for every single position in their genome.
Right, in a way that would maximize diversity.
Yeah, and that's entirely possible. That's possible, right? That's a much more reasonable model. That's what Nathaniel Jeanson has talked about. That's what John Sanford and Rob Carter have talked about, and that's, I think, where they believe their current models are.
Right.
Okay.
Do their models differ, Jeanson's and Sanford/Carter's?
Well, what's interesting is that Jeanson has his way of making a case for it. Sanford and Carter have their own way of making a case for it. And they're different. There's no overlap in the case they make. They don't really cite each other, which is a little strange.
It's completely, in each method is completely independent from the other.
Well, I mean, conceptually they're not, but they do it very differently.
Yeah,
so
in methodology.
So finding a problem with Jeanson's approach doesn't necessarily mean finding a problem. And they're dealing with different types of DNA and all that. But underlying it, the underlying model they're using is the same.
Right.
And then they both hint about the third model we'll talk about in a moment, but they don't actually go there because they don't think they need to yet.
Mm.
And that's okay. So maybe all that happens as we look at the data more, we find out, okay, this model doesn't work, and there's another version of young Earth creationism that does. And I'm open to that. Let's just see if that works then. Okay.
Right. Let's engage with the data in an honest way.
Yeah, exactly. And I think it's a real mistake to, I'm not a young Earth creationist, but I think it's a mistake to make a straw man in their position. Just because one young Earth creationist model is wrong doesn't mean they all are. But what I can say is I haven't yet seen one that really makes sense of all the data.
Right. Yeah.
Without invoking tons and tons of ad hoc miracles, but that's a different issue.
Yeah, and that's the whole crux of it, right? It's if I am of the position that if a young Earth creationist would like to invoke miracles, that's and hold that ideology and belief system, that's okay with me. My as a personal individual is when they come at it from an angle of this is all empirically supported exactly the way we're saying it.
So this is actually something I had the privilege of spending some time with John Sanford a couple of years ago, and I asked him about this, and they really are not trying to invoke ad hoc miracles. That's why they're kind of starting with something that has Adam Eve as normal people going forward, and they're only trying to move from there as they need to, because they know that if you start adding in strange things,
lose people.
It starts to look, because it's in the simplest way, you could just say that it's all a miracle. God's just making miracles all the time that just ends up looking this way. We don't know why. It just happens like that way. I mean, yeah. So I guess that's a model for young Earth creationism that could work. It's kind of a little bit like follows, or seems to make out a little bit deceptive. But you could say that, right?
Right.
But that's not what they're trying to do. They're trying to see, okay, well, let's try and build a plausible model and see if we can actually test this using scientific means. And so I'm open to doing that.
Right.
But the second model was, like I said, that, and that's, I think, where the time to most recent 4 alleles, and also we can talk about a time to most recent 10 alleles, is pretty valuable in testing if we want to think about the noetic bottleneck too. And
Right.
I think that rules it out. And we can talk about that in a moment, but I want to go through the next other models they could take.
This second model starts with the 4 unique created heterozygous alleles, but unfortunately just doesn't have enough time to generate the required diversity for what we see from the data. It fails miserably because there isn't enough time. Neither Sanford nor Carter, both of which propose the created heterozygosity that we see in Model 2, have even addressed the problem, let alone proposed a solution.
So the model after that they've really put out there, but haven't moved to yet, but kind of put it out there as like a possible place they might move to. But Reasons to Believe has recently published a little bit and talking about how they're moving to that model too, is to just accept the idea, well, maybe Adam and Eve don't actually have the same biology as us.
Right.
And in fact, their reproductive cells, either their ovaries or their testes, or maybe both their ovaries and testes, have different genomes in each cell, right?
Right.
And so then, if Adam and Eve have 20 kids, that could be 40 different alleles, right?
Yeah, they're all completely uniquely diverse, right?
Yeah, and so this does put limits because, I mean, they didn't have 1 million children. I mean, you start doing some math to see how long their lives actually were, and hopefully Eve isn't barefoot and pregnant her entire lifetime,
right.
but you can get some sense of, okay, so maybe there's some number of people. I think if I remember right, AJ Roberts, Reasons to Believe, suggested maybe up to 26 children might be reasonable, maybe.
Right.
Okay.
So it's coming up with a maximum number of children she could have, and then seeing if the data could potentially match with that.
Yeah, and so with that, then that means that if that's the case, so that means that however many children you have, you can multiply it by 2, and that's how many alleles that could be held within that first population.
Right.
And that would be different than Adam and Eve. So the first model, you only have 4 alleles at the beginning, and they kind of, 4 alleles per position. Of course, there's millions of positions, so it's really millions of alleles, but every position has only 4 alleles. And at most 4 alleles, and then that you kind of see how that spreads out from them. This is a little different because you're not really worried so much about Adam and Eve, but really their family and their family.
Yeah. That first generation then is what's being the focus.
Yeah, and so that actually does a lot to help. Actually, it means that maybe you could tolerate up to up to 50 or so, alleles that it goes down to, and that really helps a lot. But the problem then becomes Noah's flood and that bottleneck, because if you think about it, there's going to be 10 alleles.
Right.
There's Noah and his wife, who have 3 kids, but all of them get their DNA from their parents, right? And then there's these 3 other people that they marry, right? So that's 3 other people plus the 2 in the beginning. That's 5 people, right? And then you have times 2, so that ends up with around 10 alleles.
Right.
Probably in reality, it's probably a little bit less than that, but we can go with 10.
Yeah, we'll be generous.
And it turns out if that's the case, and if they all have normal biology like we expect, then the time to most recent 10 alleles isn't 500,000 years ago. It's about 180,000 years ago.
Right, which is better, but still problematic.
Yeah, well, it works for an old Earth creationist, maybe, right? So that's where it might work for Reasons to Believe, and so that's kind of, and Hugh Ross's group and AJ Roberts and Fuzz Rana, maybe that makes space for them. It's still a little weird. You kind of have Adam and Eve with this really strange biology. The way they explain it, which might make some sense, is that there's actually good theological reason to believe that that would be the case, because if they're going to be founding this population, God wants it to be, to have like an initial boost of diversity. So they're not all, breeding essentially with, they're not all inbred, right?
Right.
And so, it's a miracle, but it's all that miracle is just in the creation of Adam and Eve. It's not really anything afterwards. So, it's not how I see things, but there's some coherence to it. So I can go with that. And they're being upfront and honest about it.
Yeah, it's a cogent proposal from the position that they're coming from.
Yeah, and to be clear, we don't need this. There's no evidence that indicates this within science.
Right.
The only reason why a person would come to this is if they thought Scripture taught a particular thing.
Right.
Right. And that's okay. What we can do is say that, well, from a scientific point of view, we don't have evidence that really necessarily rules that out from what we've seen so far.
Right. There is no data set from science that precludes it, is what you're essentially saying.
Exactly. Now, what they'll also say too is people who know something about human evolution might be thinking, but wait a minute, don't we know that Homo sapiens also interbred with Neanderthals? But they're open to that too. They're saying that maybe that initial population that somewhat corresponds to what scientists means by Homo sapiens, maybe not precisely the same thing. Kind of the first with the human mind, they're kind of forming. And yeah, there's some later interbreeding from Neanderthals and all that. So they potentially actually have a very reasonable position.
Right. It kind of weaves in like a lineage for true humanity almost, and allows, once the departure from the garden occurs, potential crossover with the species that are existing outside of the garden as non-humans.
And it's interesting actually how they talk about this. This is the point of confusion. They still would call this a genetic sole progenitorship.
Okay.
Even though DNA is coming from other places. And so I was talking to AJ and Hugh and Fuzz, actually the beginning of this year, we did a big workshop with them. And what they mean by sole genetic progenitorship isn't that we get all of our DNA from Adam and Eve.
Right.
But rather that the DNA that makes us human comes from Adam and Eve.
Yes, it's almost like a genetic basis for the idea that I've at least seen and held at one point from theistic evolutionists, that Adam and Eve are the first individuals with souls, right?
Yeah, so that's similar to what they would say. Though, to be clear, some Christians would say even dogs and animals have souls too, but it would say like the first that had a human soul. Yeah.
Right. Yes, the first of being made in the image of God. Right.
Yeah, that's what they would say, and they're very, that's like really important to them. Once again, I think it's not that science tells us that their position is correct, but I don't know if the genetic evidence shows us that what their position is wrong, especially because they've modified their position in response actually to this time to most recent 4 alleles, right? Now, so we talked about 3 models. We'll talk about the 4th one, right? So the first one was homozygous clones, which is a straw man of their position, strange.
Yes.
Second one is the idea of
the classic Sanford and Carter.
Yeah, where they're currently at.
And Jeanson.
And we're actually, I would say, this is also where Reasons to Believe was too, of saying that they're heterozygous people but normal biology, right? But then Reasons to Believe has moved, and because they actually engaged honestly, I think, with the time to most recent 4 allele data.
Right.
To say, okay, well, that data really does seem to indicate that Adam and Eve probably were mosaics. So we can call it the genetic mosaic model or germline mosaic model. I think they are saying that Eve was a mosaic, but maybe not Adam. Whatever the case, there's some sort of mosaicism that's going on with Adam and Eve, okay?
That's unique to the human population.
Yeah. But then after that, it's all normal.
Model 3 starts from the same position as Model 2 with the created heterozygous alleles that are unique, but allows for enough time, essentially fixing its problems. The issue is, not only is it now an old Earth creationist model, but it seems to arbitrarily decide where to apply real science, in the sense that a large timescale is required to get the diversity from this heterozygous pair. It's fine to allow for that, but there is no mechanism to give for these created heterozygous individuals in the first place.
The 4th way to handle it, which no one has gone to yet, they've just kind of maybe hinted and alluded at it, is that there is something seriously strange and funky going on with the early human population's DNA, and something that we don't actually observe in any other populations right now. Much higher rates of recombination, much higher. There's like other stuff, mechanisms that were at play, 6,000 years ago that we don't really even see anymore among current populations. So, potentially even miracles. So no one has gone to that position yet, but that's out there.
Right, and so that's a potential terminal end to this, right? Is to gesture at that and essentially say, look, we don't know that it didn't happen, because in science, right, there's this idea of finding evidence.
Well, it depends. So if they're saying there's a natural mechanism to do that,
right,
then we should see evidence of some of the things that they're proposing. So we can even say that we can split out the things where there's a natural mechanism versus it's just like ongoing miracles. Right. If they're going to go to ongoing miracles, well, yeah, okay, so maybe that happened. I mean, there's no way.
Where do you draw the line there, right?
You're talking about that. I think everything I've heard there is deeply implausible, but no leading creationist I know about has actually taken hold of that and said that this way how things work. They've gestured at it maybe many times, and maybe there's some people who don't really know their stuff very well who think that they've gone that way,
right?
But that's not where they're at. They're actually more in that second model we discussed,
right?
And I think
that's where we're seeing a lot of young earth creationists occupy currently.
Yeah, and so we can talk now a little bit more about how the time to most recent 4 alleles and the time to most recent 10 alleles impacts this.
Model 4 is honest in its presentation, admitting that in order to get the required diversity in 6,000 years, you essentially need a biology in the progenitor couple that has never been seen in nature before. Essentially, magic biology. While it should be praised for its transparency, it's important to remember that in this model, there is very little that can be classified as empirical. And with that, the 4 models are essentially summed up. Only the conventional science model both works with the seen data and is indeed empirical, both in its initial conditions and where it ends up. Carter and Jeanson will eventually have to cop to this and move their model to Model 4, but of course that would be admitting that their model lacks empiricism and would no longer be scientific, something that Jeanson and Carter alike are very reluctant to do.
And so, I also want to put out, I'm not an old earth creationist. I'm not in the reasons to believe world and all that. I mean, I have no problem with God creating us through common descent. So it's not my view, but I do really respect how the reasons to believe folks have handled this. As when they were really presented this evidence, they spent a lot of time engaging privately to understand it, and then they even did some public stuff engaging with it, and they modified their model to move from the second model we discussed to the third one.
Right.
And what they have, could be consistent with the data. We still have to keep on looking at more, and I think they're actually very intent on really trying to make sense of that.
Well,
now, so
before we move on quickly, it seems too that we've got, when we've got a hypothesis, right, in the realm of empirical science, you're looking at a hypothesis and then you're looking at the data set, and you might get data that supports the hypothesis. You have data that could potentially preclude it, right? And then you have data that neither supports nor precludes, and acts either potentially in tandem or potentially not. It's agnostic.
Kind of irrelevant.
Yeah, it's agnostic to the hypothesis. So the first, our straw man argument is kind of out of the picture entirely. But our number 2 model by Jeanson, Sanford, and Carter is occupying the position of we think that some of the data could potentially support it, our hypothesis. But the problem is there are additional data sets that do indeed.
Well, who thinks that there's data that supports it exactly? I mean, I don't know. I don't think there's data that supports it.
I don't either, but that's what the impression that they're under is that they have data.
So Jeanson has argued that the data supports his view, but he does that because he makes a couple major logical errors.
Right.
One of them, he thinks that when you kind of go down to a single Y chromosome or a single mitochondrial genome, that indicates the population went down to that size. That's not true. We expect that to happen even if the population is large or not. So that's actually one really key place where he really misunderstands. Another place too is that he really neglects a lot of the data we have looking at pedigrees to understand the the rates of mutation, and he just doesn't even really engage that data. Likewise, he doesn't really even look at the autosomal genome. He only looks at the mitochondria and the Y chromosome. So there's just an immense amount of data. He's using less than 2%. I think it might even be less than 1% of the genome's data.
Right.
To do this, and he's using mutation rates that are just far higher than we actually observe in people today.
Right, and he's justifying that by using papers whose multiple different mutation rates that they acquired. He's almost picked the one that works for his model and kind of put the blinders on.
How he reasons to those rates is a different question. I would just say that he just ignored. I mean, like we were just discussing Y chromosome. His work on Y chromosome recently came out, and he claims that there's only been 2 published studies that look at the Y chromosome mutation rate. I forgot the exact date that he wrote that article, but even if you take the date of the article into account, that's just totally false. There is a large number of pedigree, sequencing-based studies to actually do that. He just picked a couple of very, very low-quality ones, and then he kind of did it in another analysis based on a figure. I mean, it's not the data. I mean,
so he claims they're high quality, though. What would differentiate a high from a low quality?
Well, so a key thing here is that to actually figure out the per generation mutation rate using sequencing is very hard, in that the standard ways we do sequencing have an error rate that's actually larger than the observed mutation rate.
Okay.
So if you're doing, like, for example, just 20-fold coverage, you're going to have a hard time really doing it, because when you see a change, you don't know if that's just because there was an error in your sequencing or if that's really what was observed. And that's a fundamental problem you have to deal with.
So a large sample size is just key for this.
Yeah, well, you got to get a coverage. You got to basically sequence it. So coverage is how many times you actually sequence each part of the genome.
Right. So sorry, let me clarify. When I say sample size, I mean like your in this case number of runs.
Yeah. Well, it's not runs. It's one run. It's about how much DNA. I mean, because what you do is you get a whole bunch of DNA or cells. You chop them up. You put them into a machine after isolating their DNA, and you read different parts of it, and you have to reassemble everything, right? But typically you want to have pieces of DNA that covered every single part of the genome multiple times. And that's one way how to gauge quality. I mean, there's multiple ways how to gauge quality, but one of them is how many times did you actually look at a particular part of the genome?
Right.
And
okay.
so, you can do pretty low-cost sequencing for around 10x. That means for every part of the genome, you actually sequence it around 10 times.
Okay, all right.
20 times is happening, but to really be able to do this well, I mean, you got to get up into the hundreds or even maybe 1,000-fold reads. And the studies that he picked weren't that high quality because they didn't actually have that high of reads.
All right. And you're suggesting too, and I've seen this as well. Actually, I saw you mention this on Peaceful Science and looked into it myself. The problem with that is there are indeed multiple other studies that do have higher coverage rates.
They cover the whole genome, and you can compare it. And so this is actually where it's interesting, because the math you can actually work out really easily. But if you think about what does it mean if you use a lower quality? That means that you don't know for sure what a particular point is. Like, so you actually kind of sequence it all out, and you sequence the parents too. You sequence the parents and you sequence the kid, right?
Right.
And you kind of see, okay, so these are what this person has in this position. Could he have gotten it from his mom? Could he have gotten it from his dad? If that's possible, then it wasn't a mutation. If, on the other hand, he couldn't have gotten it from his mom, he couldn't have gotten it from his dad, and it's something new, well, it's either an error or it was a new mutation.
Right.
But if it was a sequencing error, you're going to call it a mutation.
Right. And oh my gosh, right. So that's the key in this high coverage, right? That's why you use high coverage studies. It is beyond me why better.
So I think what happened is he actually focused in on those that focused only on Y chromosome.
Okay.
And ignored everything that covered the entire genome, which was a pretty major error. I don't know actually if he realizes that. I mean, maybe he knows that and intentionally did it. I think it's actually far more likely that he just didn't quite realize this issue. And so if you just look at that, that's what he focuses on. He neglects everything that covers the whole genome.
And the whole genome would, by definition, encompass the Y as well. So why would you not?
That the best approaches are going to be actually easier to do in some important ways on the whole genome than to do focus just on the Y chromosome.
Right, and that would potentially be why maybe we don't see as many Y chromosomal focus, because they're just not as robust as data sets.
The fact of the matter is, if you could for the same effort, get more patients, higher coverage on the whole genome, including the Y chromosome, why in the world would you waste your time doing the Y chromosome?
Right.
Like individually.
Right. Yeah, that really does, that seems odd to me as a methodology.
Well, so part of what was going on is well, we don't have to get into all the details of how the costing works on this, but in general, if you're only looking at the Y chromosome, you're probably cutting corners on cost.
Right.
And you're making up for it with elbow grease with text, because it takes more tech time to isolate the Y chromosome and to do things like that. But that's just not where things are right now. Right now, like, there's been some pretty large and really good studies looking at, getting large pedigrees or triads, and where you have a mother, a father, and a child, and maybe multiple children too. So you really need to have the mother, the father, and the kid.
Yeah, dyads for reasons that I plan on covering earlier in this video, and I'll probably edit a bit in this recorded section, but you can't use a dyad for these kinds of studies.
So if you're doing that, though, first of all, one of the hardest parts about this is actually getting the consent from the patients, getting them together. It's a lot of work actually to do that.
Yeah, the ethics.
If you have their data, then that's actually a major part of the expense, is getting them.
Yeah.
And now you have them. Why would you just throw away 99% of the genome to just focus on the Y? I mean, you do the whole thing.
Right.
Especially because it takes less tech time.
So instead, it would be like your M&M example, right? To make sure I'm understanding this correctly, it'd be like, I want to look at green M&Ms. I want to analyze the green M&Ms. So I can either separate out all of the green M&Ms, which takes a lot of time, effort, and money, and do it that way, or I can put the entire bag into a single machine, get separations of all the M&Ms into all their respective colors, and then after the fact, look at the green data.
Yeah, like you have like a computer algorithm that says, oh, that's the green one, so we're gonna look at it that way. Yes, that's a better way.
And the benefit of that would be now my colleagues who study orange M&Ms can also access my orange M&M data.
Yeah, so the only reason why you want to pull out just the green ones is if you have like a very small machine that can't look at a lot of M&Ms.
Right.
So, okay, we're not going to look at those, and so we're going to put that in and whatever. But the fact of the matter is, people really care about mutation rates across the entire genome.
So why wouldn't you?
And so that's, yeah, exactly. That is exactly the issue. So that's why they do that. And so there's a lot of studies that look at across the entire genome, and they measure these things. And we get very good data on this. And, we see that that there's a slightly higher increased mutation rate for Y chromosome than the rest of the genome, but not a lot. And that's just because men, as we get older, have higher mutation rates than than women do. It's a really nice study that was done on that, and it's not wildly different. It's just a little bit more. We actually have a very good handle on these rates, and they're not anywhere near. They're approximately they're about 1/50th the rate that Jeanson needs to be able to put a Y chromosome atom very recently. And even if the Y chromosome atom was very recently, that wouldn't mean the population was really tiny.
Right.
That would just mean that something else, right? So
yeah, so it doesn't really help him anyways.
Yeah, I mean, well, it could show him that it's consistent, but it's not, right?
Right.
And remember, it's only a tiny part of the genome. So if you actually look at it using the rates that we do know, Y chromosome atom and mitochondrial Eve are more than 100,000 years in the past. Okay, and we're only talking about Homo sapiens. If you start including Neanderthals and Denisovans and all these other relatives that he actually believes descend from Adam and Eve, then it ends up being farther in the past. He doesn't even include that data, but let's just set that aside for now.
Right.
We'll just look at this data.
Yeah. One problem at a time.
Yeah, one problem at a time. And so we're just even looking just at extant human DNA. We're not even going to be looking at that stuff. There's no really good reason why he isn't, given what he believes, but that's fine. And so, that pushes them back about that time. Now, if you care about the rest of the genome, the problem is you have to take into account recombination and linkage. The fact that certain parts of the genome are right next to one another, and that they travel together. And it turns out every chromosome only recombines 1 or 2, maybe 3 times per generation.
Right.
There's only a couple places that'll cross over, and you can actually see it under a microscope, where they'll do that.
Are you telling me this is observable?
Yeah, you can.
Wow, that's awesome.
You can see it. What's really amazing, you can see it in a microscope with the right staining. You'll see you'll see the 2 alleles come together, or sorry, the 2 halves of the genome come together, and then there'll be points where it's just kind of like having a crossover, like it's like a chiasma. It's called a chiasma because it looks like a cross. Right. You can actually zoom in with electron microscopy, and you can actually see the chiasma at the DNA level.
Oh my gosh, that is insane.
Isn't that cool? Yeah. So you can actually see it. And humans, we only have maybe 2 or 3. You can actually count how many under a microscope, how often it's happening. Right?
And this is consistent.
Yeah, it's like a fairly small number. Okay.
I mean, there's a range, but there's a consistent range, right?
Yeah, and there also tend to be in hotspots, occasionally outside of the hotspots, but usually they're within hotspots. There's a couple exceptions that happen in small parts of the genome, things like gene conversion and such, but they're fairly rare across the whole genome. And so even just from looking at a microscope and then knowing the size of the genome, you can actually, get a good estimate of what the recombination rate across the genome is.
Oh my God.
If you look back historically, that's how they figured it out. They didn't do it off of, they figured out the approximate recombination rate, and then from HapMap, one of the key goals of that was to actually get a more refined estimate, and it matched pretty well. And that's how we know that. In some species, you can see more recombinations per chromosome, like maybe around 8 or so. I think in some species, I'd have to find it, but to get up to 50 times that amount, to get it, to be able to get it onto young Earth creationist timescale, we're talking, like 100 recombinations per pairing. That's very hard to imagine how that could happen, just in terms of the physics of it. That would be a different sort of biology than we see in any living species right now.
We lack a mechanism, and we lack anything close to a mechanism for that sheer magnitude of recombination.
Yeah, I mean, maybe it could happen. I mean, we're talking about something that it's just never been observed before.
Right. Yeah, and that's the issue with this, right?
Standing has attempted to counter the most recent 4 alleles problem with recombination in this attempt to stay in Model 2 territory, specifically by throwing his lifeline to the PRDM9 gene. This gene is responsible for positioning recombination hotspots during meiosis in humans. The proposition Standing makes is that Adam and Eve had far more of the functioning PRDM9 genes and thus more recombination hotspots, allowing them to bypass the most recent 4 alleles problem by using heavy recombination rates. He pitched this over at the Peaceful Science Forum, where he was promptly shut down. The conversation went a little something like this. Standing proposed his idea and was immediately called out by Steve Schaffner. I had the pleasure of chatting with Steve Schaffner on the Biologos Forum once, as he was one of the authors on the landmark 2005 initial sequencing of the chimpanzee genome paper. He said, quote, "That doesn't follow logically. Not at all. There are far more recombination hotspots than crossovers per meiosis. There is no reason to think that their number has any effect on the probability that a crossover will occur," unquote. This was reiterated by Evograd, author of The Systematic Teardown of Replacing Darwin. Evograd then went a bit deeper, essentially pointing out that Standing's proposal is nothing more than a concept presented as empiricism with no data to back it up. For instance, no data exists to support 1. The notion that European PMDR9 genes are more broken than their African counterparts, 2. That Africans have more PMDR9 genes, as in their total number of copies, 3. That non-Africans are more recombination damaged than Africans, and 4. That recombination is responsible for the differences in African and non-African populations rather than the conventional science hypothesis that African populations are simply older. The analogy here would be suggesting that an ice age killed the dinosaurs. Yes, an ice age could have conceptually killed the dinosaurs, but all the data we have suggests that they weren't killed by an ice age. The data we have shows us that they were killed off by a combination of a massive impact event, widespread volcanism, and, ultimately, rapid climate change. Now, if you could show support for an ice age, then sure, we could consider it. We should consider it. But instead, in our real-world example, Standing points to data that already supports another hypothesis, that being the diversity gap between African and non-African populations, and gestures that, instead, this data actually supports this other random creationist idea. And he leaves all the details that could potentially differentiate between the two hypotheses up to, I don't know, someone else. It's not empirical, plain and simple.
But the point that he makes, which is half true here, is that it is harder to figure out how to figure out when those build the phylogenetic diagrams when you're taking into account recombination.
Right.
And it turns out that until 2014, there wasn't really any method that could do it. But in 2014, there was a paper came out. It's called ARGweaver.
ARGweaver. Okay.
Which is a program that was able to do this. And there's actually since then been other programs that have been published. There's a really great one that I'm wanting to actually apply to some data that came out just last year, actually, almost exactly a year ago. But ARGweaver in 2014 actually published phylogenetic. It's called a recombination graph, but basically it's a sequence of trees across the entire genome, and each part of the genome gets its own tree. In a phylogenetic tree, just showing you how all the alleles are related and how far back that goes, and then adjacent trees are really similar to one another, except for they'll swap different spots, and that has to do with it's like a pruning procedure. That's what happens with recombination.
Sure.
And so you can then see for every single part of the genome, how long ago was it when there was four alleles there? And,
okay.
And then you can ask, or how long ago was it that there was 10 alleles there?
Right.
And then you can kind of look at what it looks like across the whole genome. There's some that are really long, but that could be errors, or there's different ways to account for that, or there could be some a lot shorter. But what we did is we just picked the median. We said, okay, what's the median? Where is it in the middle? Just use that for now, right?
Yeah.
Maybe that's a little bit too generous to young Earth creationism, but I'm okay doing that right now. They got a lot of things going against them. Let's be generous.
I think that's wise.
And so, I mean, I do think there is legitimate critique that says that it's got to be the time to most recent 3 alleles, but not 4. But whatever, let's just go with 4. And let's go with 10.
We're giving it to them.
Yeah, and so if you take out the median time to the most recent 4 alleles lineages is 500,000 years ago. And to be clear, that really surprised and shocked a lot of people. There were some theistic evolutionists and atheistic evolutionists out there trying to say the evidence completely ruled out an Adam and Eve that far back. They were wrong. Okay. They were, once again, kind of straw manning it and didn't really have a good handle on the data. And that really showed that, maybe they were kind of not really following the data where it led.
Right.
And once again, that's not even my view. I'm just trying to be honest with where the evidence shows us when it doesn't show us. But that really does seem to present problems for having an Adam and Eve with just 4 alleles just 6,000 years ago. It seems like that would be very clear evidence that you have to move to the more mosaic Adam and Eve view, the third model.
Transitioning from model 2 to model 3, right.
Now, the problem there, it shifts now from Adam and Eve. Well, you could say Adam and Eve and say that we care about, X number of alleles. No one has yet specified to me on the creationist side how many children that they expected Adam and Eve had. So I can't really say, if they can specify that piece, we can kind of figure out what we should look at there.
Right.
But really, this isn't something we should worry about too much, because the part where there really becomes a problem was the nose flood and the Noahic bottleneck. Now you have a different problem, because the time to most recent 10 alleles, we can use that same method to figure out where that median time is. That's about 180,000 years ago. And that's not even looking at all the data. As we look at more data, it might go back. I mean, maybe it'll stay around that realm, and we'll find out. I mean, I'm looking forward to looking at that data too. And so that means now that they have a problem here, because whatever's going on that's kind of surprising and strange about Adam and Eve in their DNA. They have to do that, I think, at this point. But now they have to figure out what's going on with Noah. And as far as I know, none of them have really considered how to deal with that bottleneck with Noah.
Right.
It's not really even about Adam and Eve now. I think the real problem becomes Noah.
Right.
And that's like far more recent now in their time scale. It's just 4,000 years ago. We should be able to, I mean, we're getting more and more ancient genomes. We should be able to see the signature of far more diversity, even if they're going to propose some sort of crazy mechanisms there for like kind of spreading things out, or kind of diversifying it with lots of recombination or conversion or whatever. Like, how come we don't see that evidence? They need to specify it a bit more.
So to summarize so far, right? Model 2 is essentially precluded with the most recent 4 alleles, right?
I think so, yeah.
Yeah, that being said, if we allow for an increased level of diversity and kind of allow Jeanson, Carter, and Sanford, the young Earth creationists, to kind
of the mosaic view, yeah.
Yeah, to shift a little bit into Model 3, barring their long time scale, we're allowing them to kind of, actually what you said was Model 4. They're saying, okay, actually, we're going to go with some very wild biology so that we can sit within our 6,000.
That would be like number 4, right?
Right. Yeah. That's what I said. Within the time frame of 6,000 years, that's all fine and dandy until we reach the Noachian bottleneck, which then kind of gives everyone a wait a second. Now we have.
Well, I mean, maybe they could do it again too. Maybe but they haven't even proposed that. So like they've hinted at the other ones, but haven't moved to it.
Right.
So they can move to that. I think an honest look at the data for the time to most recent 4 alleles would have them all move to the more mosaic view. And they've already put it forward. There's no reason not to do that, right?
Right.
And I think they would also, I think that they do have to move to like a Model 4 position still too.
Right.
To make sense of that. And that's okay. But then they're still left with what do we deal with the Noah bottleneck? And they have to start thinking about that. So I think...
Do you think that could be addressed in any way? If you were...
I think it's going to be really hard to do it with natural mechanisms.
Right.
I think that they're going to have to go to miracles.
Right.
That the only real reason why those miracles would be there would be to help fit with the data.
Stop! He's already dead!
So, we've looked at a few different models, and I think it's worth kind of looking at different models and realize that just because one model works doesn't mean another one doesn't.
Right. Right.
And, if we kind of sit down and carefully step through it, we found some that work, and I think some that don't. And we haven't found anything in young Earth creationism that works yet, but there's creativity there, and it'd be really great to see, people like Nathaniel Jeanson, John Sanford, and Rob Carter, and others really engage with this aspect of the data, which they haven't engaged with yet. So far as I know, none of them have actually written about the time to the most recent 4 alleles or the time to the most recent 10 alleles. I'm happy to share the data with them and to go over it with them. They can compute it themselves. And we can even run simulations with them or for them to help make sense of it. And I think that data, really presents a challenge for the current way they conceive it, but there's some ways how they can modify it. Let's see how it goes from here.
Right. Yeah. It's and reaching out is the important thing here, right? It's extending an olive branch, so to speak, and saying, let's see where we can't get. So I appreciate so much your time today.
It's been a pleasure. It's been fun.
Yeah, absolutely. I mean, oh gosh, I know. Yeah, you can probably see the shadows like in a procession across my room as we talked for quite some time. But the time just flies by.
Yeah, I'll look forward to talking more too. And if you're a listener too, come to Peaceful Science. We're hanging out and talking. There's actual scientists there. We're trying to engage the public in a different way. And
Peaceful Science, link in the description, along with a link to Dr. Swamidass's book, which you can also find and should purchase in the description.
Hey, I'm really curious to hear what you think about that book when you get a chance to read it too.
I've got, my reading list is this high. I mean, imagine thin books, but it's very high. But man, that's going to the top of my list. I would.
Wow, that makes me feel really special. Thanks a lot.
Yeah, hey, listen, and I'm not just pandering, I swear.
All right, my friend. It's great to meet you, and hi, listeners, and hope to see you again sometime.
Oh, absolutely.
Yikes. Looks like Jeanson's up a creek, and there's no paddle in sight.
Dr. Nathaniel Jeanson, why are we up this creek without a paddle?
Because, standing for truth, you simpering sycophant, as I've told you many times before, our paddle is broken! Between you following me around like a forlorn whelp and Ken Ham texting me all the time, you're lucky I don't swan dive off this canoe and drown myself in this creek. I don't have to take this, you know. I have a degree from Harvard and But Dr. Nathaniel Jeanson, what if I were to tell you I have a way to un-up ourselves from this very creek? You have a paddle? Sorta. You want to see it? Yes. Do you really want to see it? Yes. Are you positive? Standing for truth, if you don't show me this alleged paddle this very instant, I will personally wring your neck until your trachea snaps in my skeletal hands, after which I will use your windpipe to suck down a delicious root beer float from our local Steak and Shake, home of the original steak burger. Okay, 3, 2, 1, ta-da! I use this baby all the time. The trick is to bring up unrelated subjects and pretend the argument was about those subjects the whole time instead of conceding any kind of substantial point. I bet. Yeah!
So far we've covered a lot, including a lot of the material in some videos by Standing for Truth, as well as Bad Methodology by Nathaniel Jeanson, and a lot of why radiometric dating is indeed very, very reliable. But we still have a ways to go in this absolutely monstrous video, because I'm Erica, the Gutsick Gibbon, and overkill is my favorite food. Next, we're going to discuss Standing's gross misuse of a recent paper on the great ape Y chromosomes. Now, I've covered some of this on previous videos, but for those of you who may be new, here's the gist of it. When we look at the Y chromosomes of humans, genus Pan, so chimps and bonobos, and gorillas, the human and gorilla Y chromosomes are more structurally similar than the human Y chromosome is to the chimpanzee and bonobo Y chromosome, which creationists posit is odd if we are supposed to be very closely related to the chimpanzee.
On the face of it, one may conclude that this violates the well-supported notion that humans are more closely related to chimpanzees and bonobos than any other animal. Standing did in fact conclude this, but for someone who talks a lot about genetics, he doesn't seem to grasp a very important distinction, one that was covered in the very paper he brought up, that in sequence identity, humans are still most closely related to genus Pan in the Y chromosome.
This may seem inconsequential, but I had the opportunity to discuss this topic with Crispr prior to recording, and it may shock you to know that sequence identity is indeed more informative to relatedness than structure is. I know. Shocking.
Imagine it like this. You have your favorite movie. Let's just go ahead and use the first Harry Potter movie. You own the original copy of the VHS release, as well as 2 other copies, one being the same as the original, but with the last 3 scenes suspiciously missing, cut out, if you will, from the tape. And the last copy would be Harry Potter, but with guns, as in every scene that could potentially use wands is replaced with using guns. The original film and the cut film are virtually identical in content, that is to say, sequence identity. The only difference is that the second copy is shorter. It has chunks missing at the end. When both of them are compared to the last copy, the first, the original, and the last copy are structurally very similar, that is to say, they're the same length. But the content, that is, the sequence identity, are incredibly different.
So the human Y chromosome is more structurally similar to the gorilla Y chromosome, and they are roughly the same size. But when we're looking at the sequence identity, the human Y chromosome has much more in common with the Pan Y chromosome than with the gorilla Y chromosome. To geneticists, this is something that's pretty evident, but in the wild world of creationism, this translates to...
They are desperately scrambling to make sense of the data. Easy win for Team YEC.
Which, of course, is silly. This would be akin to a flat earther looking at the revolutionary physics behind relativity and saying,
Classic. Newtonian physics couldn't answer a question, so instead of admitting it's wrong and gravity is fake, they made up new physics. They're desperate to make sense of the data. Easy win for Team Flat Earth.
Do you see how bonkers this is? Speaking of Y chromosomes, how about we take a minute and discuss these absolutely incredible population predictions that Dr. Nathaniel Jeanson is making over at Answers in Genesis that I've been hearing so much about from Standing for Truth and the Standing for Truth video conglomerate.
Starting with the assumption of common descent, we can look at the amount of differences between human and chimp DNA to predict a mutation rate, the evolution mutation rate. Starting from the assumption of young Earth creationism, which is where Jeanson is coming from, we can look at the amount of differences between human DNA from different people to predict a mutation rate, the YEC mutation rate. Comparing the two by timeline alone, we see that the young Earth creationist mutation rate is about 50 times larger than the evolution mutation rate. We can then go and directly measure the mutation rate in humans today. We do this by sequencing the genomes of couples and their children, removing sequencing errors and counting up how many times there is a mutation in a single generation. This would be the observed mutation rate. We observe that there are about 50 single nucleotide mutations per generation across 6 billion nucleotides of DNA, which is almost spot on to the evolutionary mutation rate, if a bit too low. To support the young Earth creationist mutation rate, in contrast, we would need to see 5,000 to 10,000 point mutations per generation. This is just far more than we actually observe. Jeanson makes a case, though, that his far faster mutation rate is indeed correct, and he does so by referencing two papers that he dubs as the only two comprehensive studies on the subject of Y chromosomal mutation rates. Quote, to date, 2 published studies explicitly attempt to obtain the pedigree-based per-generation mutation rate for the Y chromosome. Both studies have reported the results to be consistent with the evolutionary timescale, unquote. Of course, as we discussed earlier, this is simply not the case. The problems with Jeanson's methods of getting a faster mutation rate and thus a shorter timescale are threefold. Firstly, Jeanson's referenced high-quality studies have woefully high error bars. This is due in part to what Dr. Swamidass was discussing earlier with regards to coverage being paramount to a good analysis. Jeanson's papers have pitiful coverage when compared to other studies concerned with pedigrees, meaning Jeanson's own methodology and requirements of no evolutionary assumptions. Scally, for example, in 2016 performed a review that included 11 pedigree papers, all of which were consistent with evolutionary theory and an ancient timescale. Additional reviews have shown that by the time Jeanson released his paper in 2019, there were already countless papers on the Y chromosomal mutation rate both alone and with the rest of the genome. Second, he forgets to subtract out the error when he computes the rates, which is what gives him that high mutation rate, which in and of itself renders his work on the subject entirely erroneous. And finally, his work covers a very pitiful portion of the entire genome, just 0.005% of the mitochondrial DNA, or 1.8% being the Y chromosome. We have higher quality studies on the entire genome that show he is in fact incorrect, and he just kind of ignores this entirely.
Now, I found out about this next miscarriage of science over on the Peaceful Science Forum, which is a cool place where laymen, aspiring scientists, and legitimate scientists can convene and just chat about dorky stuff. These scientists and folks in general of all beliefs and creeds, so anyone is welcome. Link in the description. Anyways, it looks like Jeanson decided to take his Y chromosomal baggage and apply it to something. That something being the history of anatomically modern human population sizes. So what did he do exactly?
In his newish paper, Jeanson says he used his Y chromosomal mutation rate to create a family tree, and then takes all of the males from that family tree to try and estimate a population growth throughout history. He rounds this off by saying his curve matches the one we see from historians.
Now, you probably have a lot going on in your head right now, especially if you're like a layman in genetics, but you do dabble in genetics comprehension. Because here's the thing. You can't use any of that to generate a population size. Not like you can't use any of that because, you would be using evolutionary assumptions or something like that. Like you can't do it because it's a patently nonsensical method that just doesn't work based off of the principles of math and population genetics. Let me show you what I mean.
Looking at this picture here, which tracks the male inheritance of numerous human lineages, we can see that by the 5th generation, the entire population represents a single paternal line. If we were to use Jeanson's methods to extrapolate this backwards, we would get a population of 1 and not the real population, because not all men have sons.
So if this is really the case, and it really, really is, why is Jeanson claiming his method, need a chaser after that one, works and generates curves that matches those historical records? Yeah. Great question. Now I already know the answer. But allow me to lead you there, like the ferryman Gibbon of the River Styx. You won't like where we're going, but don't worry. I am here with you.
First and foremost, it should be noted that Jeanson's force-fitting of the Y chromosomal tree to his model means that many human populations diverged from one another after the year 1500 AD.
Hey pal, you just blowing from Stupid Town?
No, I'm not kidding. Jeanson admits to finding this rather shocking in an interview with the Big Daddy himself, Ken Ham.
This sort of national identity just a few hundred years ago. That's one of the shocking results of the tree.
Dr. Jeanson, I think even Christians who believe God's word and even believe the chronology taken from the Bible of the flood 4,500 years ago and so on, I think they tend to, I think most people tend to think of, these cultures just go way back and they sort of forever existed. You know what I mean? I don't think people think of that recent 1500 AD to think that before that time, you're saying all those particular groups didn't really exist as ethnicities. I mean, that's sort of mind-boggling just because of the mindset we have, I think.
And the collaborator I was working with on this project, we nearly gave it up once we saw some of these initial results because it so strongly contradicts what we all take for granted and assume.
This is pretty par for the course with creationists. They tend to take fields and place them into a vacuum, assessing them alone rather than with the preponderance of other data. In this case, the same archaeology that allows Jeanson to try and fit the Y chromosomal tree to his model via estimated population sizes tells us that the groups he supposes popped into existence in 1500 AD were around for far longer before that. But he considers the archaeology apart from the genetics and only applies the former to the latter when it suits him.
And so, in the case of young Earth creationism versus conventional science, I point to my Y chromosomal population predictions and call, woohoo! You get him, Detective Dr. Nathaniel Jeanson! I call archaeology to the stand. Using my cutting-edge young Earth Y chromosomal predictions, I posit that archaeology corroborates my work, thus supporting a young Earth. Judge, do we have to go through this again? This guy brings us here every single week with new bad methodology to try and overturn a solid scientific theory. It seriously wastes all of our time, and on top of that, literally no field agrees with him. And gravity doesn't have to come here. Right, gravity is never here doing this, and neither is germ theory. This guy just seems to have a problem with being an ape and being descended from an ape. We are not monkeys! Go get him, Detective Dr. Nathaniel Jeanson! Standing for truth, please! Judge Science, just let my witness speak. In the world of science, everyone is entitled to challenge status quo, so of course your witness can speak. If your work is good, it will speak for itself and you will be taken seriously. Now go ahead, archaeology. Tell us what you think of Jeanson's work.
Hello, my name is Archaeology.
I don't mean to offend or be coarse, but I must be honest when I say that Jeanson's work is a load of bullshit. I mean, seriously, we know that humanity is much older than 6,000 years. Egypt alone proves that. Why do you think no one not previously invested in young Earth creationism supports it? It's kind of a joke. Jeanson waltzes in here with his fucking garbage methods and force fits his work to known population sizes to try to give them credibility. It's nothing new at best and dishonest at worst. Thank you, Judge. Court dismissed. No! Stop laughing, you monsters!
But the situation is even more dire than that. Jeanson includes some graphs in his paper that, without justification, flatten out enormous population growth humps known to have occurred in human history. He flattens them because those curves violate his results. He flattens them without explanation and without restraint. He seemingly manipulates the data. Compare this to a study that was capable of genetically corroborating 100 events in human history, from wars to famine, all using evolutionary mutation rates and timescales. Let the gravity of this sink in for a moment. The study used genetic concepts and rates that corroborates evolutionary theory and indeed an ancient Earth, and that corroborated 100 known events in human history.
This is the kind of data that Jeanson needs to deal with. This is the kind of data that he's currently ignoring. The night sprawls out ahead of us, starless underbelly, rife with creationist hacks like Nathaniel Jeanson, who seemingly manipulate data all because they can't cope with being a monkey. So allow me to summarize this video.
Standing for Truth's channel frequently uses outdated arguments, obfuscation, buzzwords, and unwarranted dismissals. Radiometric Dating still is, and always was, solid as a rock. Standing for Truth's evolutionary fairy tale video stumbles majorly by relying on, among other things, glorified laymen. Jeanson's work on mitochondrial DNA and the Y chromosome does absolutely nothing to support a young Earth or creationism. In fact, it takes the light that previously bathed creationism in outdated ignorance and changes it to one of potential intentional deceit. Jeanson has, intentionally or otherwise, not addressed the innumerable papers that beat the fuck out of his hypothesis, nor has he even addressed the larger concepts of many more mathematically damning works like that of Dr. Swamidass. Moreover, so many questions remain unanswered, some of which have been asked already, some of which being new, all of which, alone and in tandem, chew up young Earth creationism and spit it out so as to not taint their scientifically sensitive palate. To ignore these questions is, in my eyes, a tacit concession. To pretend to answer them, perhaps in a snappy video titled Gutsick Gibbon Debunked or Response to Gutsick Gibbon, Gibbon Destroyed, without even mentioning the questions themselves, is also a tacit concession, and neither will get further engagement from me. To attempt to answer them in earnest, even if incorrect, will elicit a new bloom of conversation and discussion, with the caveat that the arguments themselves are new, or the pillars which held up old arguments are novel. Otherwise, I'ma head out. It's not worth your time or mine. And so, with that, I've decided to stop engaging with the Standing for Truth Creationist YouTube channel until honest dialogue can be restored. The very moment, the nanosecond that questions start getting answered is the moment I'm back in, baby. I am back in and ready to chat and discuss and have a fun, open dialogue. And it may seem dramatic, right? It may seem silly, but honestly, I put a lot of work into these videos and video responses. Hours upon hours of research just goes into making sure that I answer questions to the fullest of my ability and in a way that my audience can feel satisfied, like they've seen the up-to-date literature. I consider that my job as kind of like this proto-science communicator. I spell out my questions for the creationists to have easy access to because I don't want them to feel like they have to slog through a massive video of mine just to see what my criticisms really are in kind of a short form. And, you know, it's because I know my videos can be long-winded. And I get nothing in return from these guys but like hot air, and all that posturing must be exhausting. Especially when you could just, like, answer the questions, right? Like, you could just answer them outright. That's all I ask. Because, like I said, won't answer is starting to look a whole lot like can't answer. And you would think with all these ultimate debunkings and exposures and overturnings and invalidations and early retirements that these guys could do so easily. But you won't answer the questions. And I don't really think that you can answer these questions. That has got to feel pretty claustrophobic. And so you have my pity. You may not want it or think you need it, but you have it. Because I cannot imagine what it must be like to be a creationist nowadays in an era where fewer and fewer people agree with you every single year. And God, it can't be easy. It must be frustrating. It must be exhausting. But I can't engage anymore if the dialogue isn't going to be a two-way street where I answer your questions and you answer mine. And so I hope you'll answer my questions. I really do, because I do so enjoy our talks. But ape to ape, if you don't, well, that's all, folks. Also, like, the blooper reel is going to be me blowing out these candles. They burned pretty short. That was a long-ass video, and that was just the audio recording for the video, let alone the voiceovers. Look at all these spooky pink candles. They're really not that scary, are they? I was going for like a creepy vibe. You know, you have the skulls, and I thought it'd be cool, but, I don't know. It's kind of dumb. We'll do a cool ending thing. Let me turn this. This is what I wanted to do anyways. Okay, wait. Ooh, spooky. Alright, ready? That's all, folks. Nice.